Hey, good morning. My name is Chris Ritson. I live on Oahu. I grew up near Kailua, and I really appreciate being here. It's nice to be around a lot of people who are interested in this and have a lot of background on it. So I'm going to save you guys explaining how mushrooms will save the world and all this stuff. I think you agree with me that this is a really important subject. So. I am going to give you a little overview of my art in the end. I want to give you a little background, though, so I'm going to talk about a really brief history of art and a lot of it how it has to do with mushrooms here. So before we set out, there's a division in Western art and non-Western and folk art. And what we start considering is like classical art in the Western culture very clearly has negative associations with mushrooms. Much later, this was described as fungophobia by the British mycologist William Deslil, and it's still very present in our society. I deal with it as an artist very regularly, and I'm sure a lot of you working in science and other sort of researches or medicine experiences too. But it defines a lot of the work people were making. And then in non-Western folk and indigenous art, we see really different perspectives, usually linked to mystic, sacred, or medicinal qualities, and also as just ornamental motifs on a lot of things. So as far as I know, this is the first artistic representation of mushrooms. I'm sure you guys are pretty familiar. It's over 9,000 years old. It's a petroglyph in Algeria by a group of cattle herders, basically, the culture. Terence McKenna is very famous for claiming these are psychedelic mushrooms and a shaman. And I think it's very possible if they are, taking huge flocks of cattle around that we could definitely consider these might be psilocybins. But this is a very favorable, this is like a towering creature over what were also pictures of cattle and hunters and such. A little later, we find this up in northern Russia, these mushroom-headed people, and it's another consensus that these probably represent Amanita mushrooms. They were reindeer herders and also whale hunters. We also have the Mayan mushroom stones, which are associated with rituals, psychedelic rituals. And we're finding them from 3,000 to 1,500 years old. I'm not as versed in Eastern Asian art history, so this is sort of overview of some of the motifs you're going to see. The fellow on the right is sort of the godfather of Chinese medicine. There's a very famous picture of him depicted with a Ganoderma mushroom. But otherwise, you'll see him on motifs in vases, you'll see him in paintings. Lingzi are very, very favorable mushrooms as an ornament in Asia. And then before the 15th century, we don't see a lot of depictions. Greco-Roman art doesn't express a lot of mushrooms, although they were probably pretty present as a food source in a society. Leading up into the Dark Ages, there's very little representation. Most of the art we find there anyway represents religious identities, Christianity, such. But after that, when we start seeing classical paintings we see a lot of mushroom paintings, usually in these sort of still lifes. They're very famous in Dutch still lifes, German, Italian still lifes, but I want you to really pay attention to how horribly these are. They're usually alongside snakes and toads and rotting flowers and such, but it is interesting because people are departing from painting perfect flowers and perfect mountainscapes. They are looking at a little bit more macabre subjects. This one I love. The mushrooms are actually rotting a little bit and eaten by bugs. And you see these vipers on the background, this sharp nettle. But then if we can kind of fast forward to the 1800s, Victorian science and the Enlightenment, the sort of culmination of the Enlightenment, leads to these botanical drawings, which I think changes the perspective of society a lot, and they're no longer seen as poisonous or dangerous. that they might be organisms worth studying. And there's a huge bounty of these sort of drawings. They're a lot of fun, very detailed. But what I think is interesting, these sort of fantasy paintings start happening. Fairy rings, that's where we get this term from, and there's a lot of paintings as such. Lewis Carroll. I mean, maybe there's some Orientalism here with the exoticness, sitting here smoking his hookah, but still representing wisdom, representing these other things. This Art Nouveau painting here. I just think it's really lovely, and we're seeing a shift, a very slight shift, but they're not just evil anymore. I'm particularly interested in using mushrooms, living organisms as art, and I think it's really interesting that Alexander Fleming, who discovered penicillin, also on the side made these microbial paintings with bacteria and yeasts on agars. And yeah, he might not have been the best drawer, but the idea was really there, and I think it's neat to see such a famous scientist playing around with science in ways that aren't typically what we think of as productive, and that these things can be used for purely ornamental purposes, which has a lot of value. I mean, that's what we're talking about with a lot of this historical art. There still is a lot of remnants of the evil in mushrooms and the darkness in mushrooms here, and you can see it being used as propaganda by the Germans. In the Reinhardt regions of Germany, where a lot of the Ashkenazi Jews lived before the Holocaust, mushrooms were very present in people's diets to the extent that Germans called them Jews' meat. And this book was very widely distributed to children in schools. And it's about how Jews can be understood as poisonous mushrooms, and they're very hard to distinguish, but it's very important that you know what's going to be, necessary in weeding out in a culture. And, it's very sad, I think. It's very, very expressive of people's misunderstanding of nature and society here. But then I'm going to kind of jump into modernism. In the late 1800s, leading up into pretty much the 1970s, people start experimenting with different forms of art that are not purely representational. We have a Mondrian on the left, which kind of represents minimalism, which people will be doing artwork that is purely as minimal as possible, and cannot represent an idea or a concept or a sense of perfection. Pollock in the middle, these abstract drip paintings, and Pablo Picasso on the right, who's the drip paintings are purely abstract. Can you make a painting that doesn't represent anything? Picasso is using different forms of vision and understanding, and that's coming out of Impressionism, Vincent van Gogh, and things like that you might be more familiar with that are leading into these sort of abstract modes of modernism. The thing about modernism, though, there's very little representation of actual things, so I find it very hard to find mushroom imagery here. I did find these Alexander Calder pieces. He invented the mobile. If you have children and see a mobile spinning around, you can kind of give credit to this artist. These aren't his most iconic works, but they all have mushroom in the title, so I appreciate that. Around the 1970s, we have what's called postmodernism, and it is a very large departure from the history of art. We no longer can think of artists as purely geniuses, purely representing ideas that are universal to humans. We start seeing context being important, where you're coming from, why you're doing it, what's funding you, the privileges or hardships your society, your ethnicity, your cultures have faced to get to these points. And in that regard, you can't have a abstract painting in any way. It's informed by the painter. And so a lot of art now will be about the artists themselves. I'm just going to show a few really iconic works that stand out to me. This is Richard Prince's early work. He was dealing with appropriation. On the left is a Marlboro cigarette ad, and on the right is a piece of art by him, which is basically the same thing. He's taking that image, appropriating it, and putting it in a different context. And whether or not you believe that's art, it draws a huge controversy about thinking about whether that's art. And a lot of his fame comes from being sued so much, and he's been very successful with it. This is Joseph Beuys. He's a German artist. This represents, I think, performance art, or people are doing work just by the movements, the associations of their bodies and spaces. And for this work, he flew to America, got off the plane, got in a taxi. They drove him to this room, and they locked him in this room. And in this room was a wild coyote. And he spends about a week in here. He has this wool blanket, a staff, and there's a bunch of piles of straw in there. And he develops this relationship with the coyote that's both violent and sort of by the end, you see him on the right, they become friends, and they're sitting looking out at things. And so this is really about the sort of relationship you can have with the wild, with foreign cultures, and within the context of a white gallery, it's very revolutionary. Cindy Sherman starts creating these photos in the 1980s, and what she's really famous for is inventing the selfie. Up until this time, there was a photographer and there was a subject. It's the same with a painter. She turned the camera around and starts considering what if the artist is the subject of their own art. And this is different than a self-portrait in a lot of ways, because she's putting herself in these really strange situations a lot of the time, and she's mirroring other tropes of art that are very, very problematic for women, I think. And so the things she did really affect us now, and I think give us a lot of agency for the way we think about ourselves and the way we can do a selfie and, validate it just as much as having someone else take that photo of us. Jeff Koons makes these really absurd, ironic sculptures. Maybe you've seen them. Some of these are humongous, 40 feet tall. They're made out of metal. And I think his work, to me, troubles me a lot because of the amount of resources necessary in them. But they are some of the most popular contemporary collected pieces right now. And they do look very cool. In the back, you see a porcelain sculpture of Michael Jackson hugging a monkey. And it's some of the most absurd stuff you'll ever imagine being turned into fine art. And that's the appeal of it, that subjects don't have to carry the sort of weight they did. And we can give value and representation to these cheaper parts of society. They're more mass-produced. Within this, a genre starts to develop called bioart. I'm just going to talk about one of the most famous pieces by Eduardo Kac. He's a Spanish artist. He usually works with European laboratories, and for this first one, he made a transgenic rabbit that had a fluorescent protein from a jellyfish. Why do that? It's not the sort of purpose we see in genetic modification, but is that act there representing the same sort of act as creating a painting or creating a sculpture? At the time, and still now, it's very controversial whether or not he should be doing this. And it's also very questionable whether or not he actually did it. This is about all we know. He has done he's very into stem cells now, and so are a lot of bioartists. We have other bioartists doing stem cell surgeries on their own bodies. I don't think I have a picture. Stellark's famous for he's implanting a third ear in his arm that will be hooked up to the internet. But also at the same time, there's this genre called generative art. And that's where we have an autonomous system creating the art, something non-human. So this piece here is just a box with some water in it. And depending on the temperature and the humidity of the day, the condensation will change. So it's constantly changing, but has no input from the artist. So we'll also see algorithmic art. We'll see just equations that are given to a gallery, and the gallery will fill out that equation and draw the lines on the wall. And very quickly, generative art became about computers. And I think we're very familiar with generative AI right now. It's a lot of fun. People are really questioning whether or not some of these paintings are allowed in a nicer context. I think these Dali ones are some of the earlier ones, and they're very freaky because of the way they are designed, the way they come out. I found this one on the internet called Neurosurgeons Putting Ramen Inside a Man's Head as a Joke. So you put that prompt in, and the computer comes up with 9 photos. Other programs now have gotten very realistic, and people have a lot of trouble determining whether or not it is human or AI. So I think generative art is going to be really important for the future of how we understand imagery and literature and most other forms of human output. So postmodern art sort of dissolves in the 70s, where it becomes more important to look at narratives of post-colonialism and globalism, things like that. It becomes very complicated, too, because we can't really define the present moment. So I picked out some successful contemporary artists who are using fungi. Zhaojing Yan is from China. She does a lot of work with classical Chinese representation in more contemporary views. And these are made out of Ganoderma reishi mushrooms in a mold. The blocks are mycelial. And she does get them to fruit, usually in a gallery. I'm very impressed because they're usually always covered in spore. But I think she's done some really good work. This is Noor Mobarak. She got a lot of attention for these beach balls full of, I think they are turkey tails. Where is she? I think she's out of New York. And then, there's quite a lot of work that's gotten notoriety for just being mycelium blocks. Jamelia Mika one, she did these in a they're in a misty room in a gallery. A lot of stuff you'll see in galleries doing a lot of myco materials too. They'll be representing spaces and structures that are made out of mycelial blocks. And I guess because there's not a lot of understanding of what they are yet, they are very frequently shown in different versions, for better or worse. Okay, so I'm going to get to my work now, which is really informed by those last 2 generative and bioart genres. I started making these paper sculptures. I was working in a video rental store in San Francisco, and we had all these. They would give us posters every week, and we'd have these piles of posters afterwards. So it's not quite origami, but it is, I think it's called kirigami. It's cut and glue sculptures, and they're all using bodies from the representation in popular media, popular movies. Later on in Hawaii, I was commissioned by the Honolulu Museum to make this video that is, it's a long loop. It's a stop-motion 3D animated digital movie. The stop-motion, it is a narrative of a coral reef, and all of the human representations you see are from the media that's distributed across Waikiki. If you've been there, you see there's these newsstands full of tourist magazines, lots and lots of people in them, tourist magazines and real estate magazines. So these bodies are put together and made into a reef space. But at the same time, I've been really interested in trying to produce a generative art form, and that was with crystals originally. Crystals really were interesting to me because immediately I didn't know how to work with living things. It's hard to consider how to do something in a space or a gallery with something living, but a crystal is very hard sometimes to distinguish from something living. They reproduce, they can change, they can sort of navigate spaces and adapt to the environment of the room or the environment of the structure they're growing off of. And I got particularly interested in sugar. It's easy to work with because it's non-toxic. It's definitely not archival, but I don't know if you can grow incredible crystals out of sugar. And this is grown off of a boar's skull. And if you live in Hawaii, I'm sure you've experienced the trouble of boars in the forest or in your farms, but they also have a really important role in the history of agriculture here. And this kind of is about the way that the sugar plantations moved right over that. So sugar you crystallize by making a supersaturated solution. It's basically like if you've ever made syrup, you melt the sugar down into a little bit of water, and the hotter it gets, the more the water can dissolve. And then when the water cools, the sugar precipitates. This is how a lot of crystal growing works. The material precipitates, and as it precipitates, it reforms into its atomic structures. So the more saturated you get it, and the slower you cool it, also depending on if there's a sort of seed crystal for it to begin to grow off, the cleaner and bigger the crystals you will get. So this was fun, but it's very hard to sell work like this. As I'm sure you can imagine, the ants love them too. So I was looking at a lot of other crystals. A lot of stuff is very poisonous. I did work with some fertilizers, more as chemical pieces and things like that. But this is an element called bismuth, and it's a metallic heavy metal. It's denser than lead, and it has this incredible structure that sort of represents like a tesseract, a fourth dimensional shape that moves into itself through time. It's a really, really strange concept to me, but they also oxidize with this beautiful rainbow color. The rainbows you see on it's actually a rust that forms instantly, and they are grown similar to the sugar. You melt the metal and slowly cool it, and if there's a catalyst, which will be where these crystals have grown off of I use porcelain sculptures, the crystal will form, and the slower it forms and the way it moves, all this defines the crystal. And you start getting some really fun shapes. And I guess I'm questioning, I'm not making these sculptures, I'm starting the sculpture, I'm seeding the sculpture, but the material is ultimately deciding the way it's going to look. And a lot of these are overlaid in more traditional forms of art. Porcelain sculptures are usually religious or, classical or very risque in a lot of ways. And so all this work, I should mention, was commissioned by an app that used to be called VSCO. I don't think it's around anymore. Okay, so this is my first foray into living art. And what you see here is a piece of glass from underwater, covered in something called coralline algae. Coralline algae is a calcareous algae, encrusting algae. It means it deposits a calcium skeleton onto the surface where it grows and within its cells. And we see the reef. We talk about coral, the reef being made out of coral, but in fact, quite a bit, the majority of it's actually made out of coralline algae. And coralline algae deposits over and over itself in a healthy environment. It is what actually fuses all the rocks together. And it sends out signals letting coral know where to land and where to start growing when it's in its planktonic stage. And it is very much affected by climate change and ocean acidification, temperature changes, just as much as coral is, and over competition with invasive algae. This work was commissioned for the first Honolulu Biennial, and it was designed, what you see is an aquarium system, a saltwater aquarium system with these panels hanging in it. And it is designed in such a way the environment is controlled to be slightly warmer, slightly more acidic, and slightly higher nitrogen than the ocean is normally found. So it's basically an apocalyptic situation in the ocean. Something like this, we're going to expect most of the coral to die. We're going to expect a lot of problems happening that probably going to be irreversible. So these panels are in here along with this detritus from the ocean. Hopefully everyone knows you cannot collect rocks or coral from the ocean, which is where coralline algae typically lives. But I went out and collected a lot of these glass bottles and pieces of garbage, which is just covered in it. And slowly these organisms spread into the water. They scrape off. They do just particles scrape off, and they sort of colonize on the surfaces. And in the end, we have these panels that are dried and preserved, and the skeletons of these organisms remain on them. And what is so valuable about these organisms, these representations, is that this is an artwork that looked into finding solutions to climate change directly. And this is the representation of the literal organisms that are going to survive this. So in a grand scheme, we could culture these guys out before they were dried, and have, a team of coralline algae that are much more suited to problematic environments. But of course, this is an art piece, so it's just really representing that. And I did like the way these turned out. And these are strictly archival. These will last, outlast hundreds of years at least, hopefully forever. But that got me thinking a lot about how to make the whole process sustainable. I just want to kind of share this description I have of art. Art is not a commodity. The context and materials of an artwork are just as important as the subject it represents. Instead of interpreting a painting through the image it's depicting, imagine it for what it is. A collaboration between chemical paint companies, factory workers, shipping companies, and retailers. While we increasingly experience media through the digital representation and the influence of massive corporations, it's more important than ever that we remember to consider the physical networks necessary for the creation of images. So working with some of these larger galleries and events, it becomes really apparent immediately that they are just, there's no foresight into what's going to happen afterwards. So much money is raised to do these exhibits that will last a month, a couple months at best. Walls are painted, new lights are put in, walls are actually built. Spaces are completely taken over just for this time, and there's no insight into, is this going to help the culture? Is this going to do anything more than give us, the artistic experience is very important, but it's kind of stripping us of the relevance and the actual reality of it. Aside from that, moving art around is very expensive and is very wasteful. Even in Hawaii, we'll see humongous metallic sculptures shipped in. Most of the public art you see that is a metal sculpture was actually maybe made by Hawaii artists, but cast on the mainland and shipped back here. And a lot of times it's sent back, and its stuff is sent all over the world. And it is good for people to experience this, but I think during the pandemic, we thought about this a lot and how we're going to experience art. Are we going to be going to museums and packing into art openings still? Is there more relevance in having experiences that are more intangible? I'm not sure, but I was questioning this stuff. And so it led me to look for materials that were more sustainable, more smaller impact on where they came from and what was led to creating them. And that led me to mushrooms a lot. This I included because this is the very first grow I ever did with the Ganoderma. This is a Chinese reishi, this species, Ganoderma lingzi. And I'd read if you keep them in a poor environment without a lot of air exchange, they make these antlers like this. And I just thought it was so fascinating that you can adjust the control of a mushroom and have such a drastically different shape than how you would see it in the wild. And this was the first exploration I did. It was what happens if you block it, if you create a two-dimensional space for it. And this doesn't look like much, but it is a very different way of seeing the growth of something. And so this is growing up, hitting a piece of glass, and spreading across the glass. And I think just as a gesture of our pushback into art and our experience with nature, we often put up these boundaries, and especially with nature. It's very rare for somebody to actually involve themselves. I'm sure not with you folks if you're here right now, but to actually get out on the ground and see a mushroom and consider how it's growing and its relevance is very different for people. So in a lot of ways, that glass wall is the society, and this is what I hope to kind of bridge with it. So I'm from Kailua. I now live with my wife on Pu'uohia on Oahu, which is usually called Tantalus. It's a cinder cone on top of the Ko'olau right above Honolulu. And I think it's just, I'm going to talk a little about the environment there. It's a very, very special environment. It's a cinder cone. I believe it's millions of years younger than the main Ko'olau range. So it popped up out of the side there. The soil is very different. The topography is very different. At the top in the center of the cinder cone, just less than 100 years ago, probably used to be a lake, a duck pond, that was reserved for ali'i. There was nobody lived up there. It was only for bird hunting, bird catching, for EEV and feathered birds like that. And it was a very, very dense native forest. What happened after the introduction of cattle was that it completely got stripped. The cattle's in collaboration with people harvesting sandalwood. This is for the sandalwood trade that was picked up often by whalers and often by people traveling the trade winds from the mainland to Asia for trade. Stop in Hawaii, pick up a lot of sandalwood, and they completely deforested the hillside. You see pictures around the 1890s of the hillside, and it looks like a desert. And so what was happening to Honolulu is the topography of this, I should also say, collects rain in a different way than most of the mountainside. The clouds condense, and it's one of the wettest places in the whole Ko'olau. And that water collects, and it becomes one of the major aquifers for Honolulu. They pump a lot of water out there, and that was all just flowing down into the streams. They were having mudslides. So what happened was actually the federal government of the U.S. instituted a program there. This is maybe just like 10 years before the overthrow. And this was called the Experimental Agricultural Research Station. And in an attempt to reforest, they said we're also going to experiment with what crops are going to work out in Hawaii. And Tantalus this area goes from pretty much sea level up to about 2,000 feet. And there's so many mountain ridges that we can plant something on north, south, east side. And we have all this different sort of soil. We have different temperatures, different humidities. I mean, there are areas that are on the south side that there's cactus growing. So they planted all this really strange stuff that didn't exist in Hawaii. And if you go up there now and hike around, it's really interesting because you'll walk through patches and all of a sudden there'll be a different tree and then a totally different kind of forest. And this program never worked out. It dissolved. They never applied to research, but what they did was introduce some of the most invasive trees we have on Oahu. So two of those is bamboo and strawberry guava was introduced up here. And just, it has grown wild, it has taken over. But there's also a huge spectrum of introduced trees. So I think this is one of the wettest, most alien forests we have on Oahu. And because of that, I find a lot of interesting mushrooms. And I learned to culture them. Started very simple. If you're ever interested, don't be put off by the need for a lab. I had a Tupperware container for a clean room, pressure cooker. That's all I needed. I mean, I wasn't even sterilizing my sawdust. I was just pasteurizing it with boiling water. Not the highest success rate, but 1 in 5 bags would succeed, and that was enough to start getting really addicted to growing them. Looking at species that preserve better was my first intent, and going through cloning species and finding really aggressive ones that made it easy to grow, made it easy to avoid contamination, could fruit outside. The one before is a mycoporous species, Mycoporus affinis, I think. This is Pycnoporus sanguinis. I think if you guys were here yesterday, you should have seen them on your hike. And those are two that really led me into this. I will clone them. Usually that little clip could come from a mushroom. This is just a old agar dish. It's being reconstituted into a new one. It just takes a very small piece to start this all. And that culture, some of these cultures has lasted me like 10 years. I still am pretty old school. I use jars. I pre-pour my agar and sterilize them instead of Petri dishes, just because that's what I have, and it's been working really well. And here's some different species. The orange one's a pycnospore. The yellow one, I don't know what it is yet, but while we're here, I'll talk about it later. I just want you to notice in the middle is where the little culture started, and then there's a disc around it that doesn't look like anything, and the further out it goes, it turns yellow. This is a really different fungus for me. I guess I'll show it to you later, but it doesn't start creating mycelial until it gets off of the medium. And even when you have it in a grow bag, it coats the side of the bags more than it coats the material. Now I use a HEPA filter that I made. I use bags, sterile bags, and the more you supplement them, the more interesting your stuff is going to look, the less it's going to look like what it grows in the wild. And that's been really, really fun. This is a block of what the pycnosporus looks like. I'm just getting ready to fruit. I will grow them out like this. Before this, I will break them up. This one I've left. A lot of them I will leave as tests to see what supplements are working better. Most of these photos here come from a grant from the National Endowment, distributed by the Hawai'i Museum of Art. So they funded me to basically do research on how I can produce larger works, I can produce more sustainable, faster works, and get a sort of timeline to break down exactly how long it'll take to go from, culture to growth. I'm working on two sort of styles, a two-dimensional style and then sculptural styles. This is a little picture of a corner of incubation room. On the top, there are sort of masks I've been doing. I'll show you what those look like in completion soon. And on the bottom, these are more akin to paintings here. These are more of the masks using some really low-tech technology. You see they're just in Ziploc bags. And these are just plastic molds. The plastic mold, you can use a vacuum form and basically take any shape. This is being utilized with packaging a lot. If you're familiar with myco-packaging, you could do this for the shape of your wine bottle and have a totally biodegradable form of styrofoam. So after the incubation rooms, I wanted to find a way to grow them outside and to fruit them outside because, let's be fair, humidity, machines, and lighting do take a lot of resources. And I am culturing these all as close to the environment as I can. I live up there. I farm flowers. I farm heliconia, ginger costus, and it's a very wet, very living environment. This area is situated next to our catchment tank, and we have enough rain. It is pretty literally overflowing, so it's basically by a riverbed here, and it is covered by a canopy of, I think these are Richmond's red heliconia and Japanese ferns. So I think the supplementation of oxygen here has changed things a lot for the way things have grown, and I want to encourage anybody looking into growing edible or medicinal mushrooms to consider supplementing with living plants. Mushrooms breathe oxygen, and plants give that out. A lot of these I will just do test batches. Again, I'm just exploring different medium, different supplements. I haven't said, the point of this really originally was that we do a lot of clearing, and at the top of the area where we live, it's mostly guava, there's a lot of bamboo, but there are also these relics of the native forest. They're koas big enough you can't wrap your arms around them. There's ohia, there's kohiko, there's nampaka, and these things are just choked out. It's it's unbelievable. You'll see them just scraggly, hardly alive, just strangled by guava. And so as much as we can, we try to clear those guava and give these guys some space to grow back, and they do, and it's really satisfying. But what are we going to do with all this wood? Is there something more valuable than just letting it compost on its own? Well, what if we compost it with mushrooms? What if we use those mushrooms to make a commodity that's really valuable for symbolic or even monetary purpose? And so that process, going from a material that actually helps the environment, that improves the process of our relationship with the environment, to produce an artwork, is really the goal with this. Here's some more samples. On the left, you can see I'm experimenting with how to do shapes on a medium. And basically, you can just get a plastic medium or a reusable metal or plastic medium to grow the shape, and then expose one part to the air, keep the other part away from the air, and stuff will sprout right out of that. This is Pycnoporus on the left, and there's a local Trametes turkey tail up in the corner there. There's a kind of larger spread of that area later on. You can start to see some of the sculptural pieces with the masks I grow. Down on the right is what is that? It's a jelly fungus. The young mushrooms are incredible. It is hard to preserve this color, I find, but they come out really strong, really bright, and when you see them, it almost doesn't look real in real life. It's just such a vibrant color that really doesn't fit with the rest of the greenery. This is a really cool mushroom. It is incredibly aggressive when I grow it out. It took me the longest time to culture it because when you put it on agar, it sends out these little filaments, and I kept thinking it's a contaminant. And then behind those filaments, you see the orange mycelia. So I just couldn't get those filaments out. I just kept thinking, this contaminated, this culture. But after a while, I let it go, and that stuff it tears through the agar, those little filaments, immediately. Like, just within 24 hours, it'll spread over the whole thing, and it just kind of is laying the way for the thicker mycelium to go. And I don't see, a lot of times when I clone stuff, you just see one strain go out. It goes out all evenly, little rhizomorphic forms, if it's that kind of mycelia. But, this one, it seemed like it has a very different relationship with the environment it's in, and that was very valuable for this application. It's also very hard. It is used in technology, biotechnology, in humongous liquid culture vats to break down fibers for paper. Usually it creates one of the strongest enzymes to break down wood. So instead of using bleaching agents, a lot of times the mycelia of this fungus as a liquid culture will be used. And then these are sort of where I arrive. These are mushroom paintings. They are about 3 or 4 inches thick, and this is where they look like at the end. They can be hung on the wall, and what they really are doing, in one way they're representing the environment. They change and grow depending on the amount of rain we get, depending on the temperatures, depending on the airflow. But in a lot of other ways, they are just defined by the plans of the mushroom, by the mushroom doing what works for it. And it's really cool. It changes over a month or two, this fruiting process. It'll go from really simple pins to these really, really elaborate structures. Here's a sort of close-up of it. And like I say, it is so bright when you see it. It doesn't look real. And they're very stable. They're very thick. They're outside, so maybe we have bugs, we have slugs, we have frogs, we have rain, we have dry days. I've gone through dozens and dozens of species. I've gone through probably dozens of strains and landed at 3 or 4 different strains that are just real keepers. And like I said, these are all picked up pretty much within like a 2-mile radius of our property that we live on. And I do think the supplementation really helps with oxygen. I'm going to show a couple of the different panels that have been a result of this. You got to decide when to harvest them. You have a younger one on the left, an older one on the right. And this is just a little, I think, this is one of the larger ones I got out of a single 5-pound block. Definitely a good supplement. Yeah, and so these are for people with fungophobia. If you don't have it, you do now, hopefully. Dentures and glass eyes are stuck into those little masks you saw. People, when I tell them I work with mushrooms, like half the time they wink and go, "Oh, what kind?" Or they just go like, "Oh, don't you get sick?" Like, it's so prevalent how scared people are of them. And, I tell myself it's because they're really powerful, not just psychedelic, not just poisonous, but, people are afraid of things that are going to really be powerful and have a lot of strength to them. And I guess that's kind of what these characters are. This is just a piece showing kind of how they're grown. They don't have to be square. They can be any shape. Here's a different strain, a different time they were harvested. And these are all life-size. Okay, I'm just going to wrap up by showing some of the other cultures I've grown. This is microporous. This one is incredible. The last project I didn't work with it as much. It's very slow growing compared to other polypores I grow. It creates one of the thickest sort of mycelial mats as anything I've grown. So if you've heard of the mycelial leather, people making slippers, stuff like that, this is the one I would go for. It is so strong. It is so thick, but it is a little slow, so I probably need to go back to that and find some better media. It also really loves, I don't know if there's an algae or what, but it loves living with this green stuff that grows into it. I've seen this on turkey tails too, but it has some sort of relationship with an algae eventually, and the more it does, the more it gets these kind of hints of blue in the fruiting bodies. There's a little close-up of them. And they're very stable, too. Once they grow, they're very thick, very, very much like a woody sort of feeling. I don't know about applications. I've heard this one is medicinal, but it is introduced. Where I live, it grows off Java plum mostly, but it's pretty readily will colonize any block. This is the largest piece I've grown. This is Tramates. This is the turkey tail, local turkey tail. And it's about 4 by 4 feet. And this one was done really low. This one was done before these orange ones. Really, really low technology here. I think this one didn't use sterilized bags, just used Ziploc bags with boiling water poured in to hydrate the wood and grain spawn. And this is what they look like when they start to sprout, these little white spots. And then once they start forming caps, I would have brought it outside. This is a culture I started to work with, but have kind of put on hold because I'm not sure of its parasitic nature relationship. It is the local Ganoderma, and it'll grow huge conchs. In Tantalus, we usually find it on koa trees, but the cultures I took came off of pine trees, cook pine trees. And this is pepeao, the ear fungus. It hydrates very well, and it dehydrates very well too, but it is one of the more alien ones too that I feel that people interact with and experience. It's very jiggly and jelly-like, and it makes fairly strong blocks too. And this is my most favorite ever right now. I don't know what this one is. It just popped up on a tree one day, and it actually popped up as like encrusting fungus. I didn't know it would make these plates at all. It's bright yellow. I'm just gonna, if anyone knows what this is, please let me know later. Got these sort of tooth-like spores, and that was it. That was the original one. pretty recently, I got a hold of, oh, this is what it grows like first. I didn't think it was going to be worth anything. I opened up the blocks I was experimenting with, and it just covered the area that was exposed with this weird spore stuff. I don't know what it was. I was like, okay, it took a long time to clone this. I'll put this one on the side. I left them out there. And, in a few weeks, the mushrooms I showed you pop up like this. And it was really cool because it's not something you see in the wild, and it is sort of testament to the interesting things you can pursue with ornamental mycology. Whether or not this has applications as a medicinal mushroom, it is just really fun to see the ways you can interact with it to make an experience with a human different. So I did get one mask out of this. And it's very leathery, too. It's just like any of the other polypores you've probably experienced. But what was so cool, after I finished this, I got a hold of a blacklight flashlight, and I went out, and I found this old colony here that had started to make brackets, and it just glows at night. And I'd never seen anything like it. Also, that little blue spot on the left is another, I'm waiting for it to grow out. It's some sort of polypore. It's probably the Ganoderma, but I'd really like to pursue this. I think there's a couple other fluorescent fungi I found around the area. I think there's a lot more. Hopefully we can pull out the flashlight and look at this stuff tonight everyone's found. But, you're in the forest, it's pitch black, and you shine your light around, and all of a sudden there's this glowing thing. And the light doesn't really light up anything else. And it lights this up. I never knew that, It's really interesting. So, okay, I'm just going to wrap this up. I think I've heard some really good call for a fungarium in Hawaii to preserve specimens for the scientific community. But, I had experiences growing up here on the windward side, and one of my favorite things to do was to go to town, to go to the Waikiki Aquarium, to go to the Honolulu Zoo, to go to the Lion Arboretum. And we have, world-class aquariums, world-class gardens. It's something tourists love to go to. I think it's something children love to go to. And I really think Hawaii should be the first place to have a living fungarium we can go to. We can see mushrooms growing. I think we should get behind this. And I think it is really for the youth. It's for the next generation. And as a child, I lived by the ocean. I spent most of my time there. I lived in the forest. But it's different when you can get right up to an organism. It takes a lot of time, patience, and as a young kid, you can't go out into the forest and look for stuff. This could double with a lot of purposes. It could be an active farm. It could be, these little stores that sell stuff do quite well in them too in these places. People have weddings at the Waikiki Aquarium. As a florist, I do work with people who want mushrooms in their floral arrangements. I think the more this perspective opens up, the more people are going to want to interact with them. And I don't think this would be a very complicated process. I thought about it a lot. I would probably first start talking to botanical gardens to use mushrooms that are already on the property, but I think if you get to a really large scale and you want people to interact, you do need to use a closed system. So I think a model more like an aquarium, less like a zoo or a botanical garden would probably be the best choice for the public. Obviously, a large tank like in the back where you have an entire farm's worth of mushrooms would be the most overwhelming and interesting, but specimens as you walk through, little things. I know Hawaii is for good reasons, we don't import mushrooms. We're not supposed to import cultures. A lot of people do, but I think there's enough focus and interest in the stuff here that it would give us a lot of value to learn how to grow it, not only to collect it. And like I say, for the purpose of mycomaterials, you learn so much by just putting something on agar that you wouldn't by looking at the mushroom or by doing chemical analysis. The structure of how it grows is totally different experience. And aside from these sort of tanks, we have to have glowing mushrooms. I think that's really what's going to get people, whether or not it's bioluminescent or the fluorescent fungi I was showing you. My experience of that was very humbling for experience in nature. And I think if I was a child, for my child, I would like to offer that to other people. So hopefully as I get this project rolling out more, you guys can all be a part of it. And if you have any interest in it or any advice, I would love to get that. Please find me. Look me up on Instagram. That's where I share most of my new work. If you're ever in Honolulu and need some flowers, we're up at Tantalus, called Tantalus Botanicals. And thank you. I really appreciate that. All right, if anyone wants to ask a question, too. Yes. Yeah.
Have you noticed in the composition of your substrate that you're using to grow, what you add in there, like nutrient supplementation, does it affect aesthetically any of the pigmentation coming out or the hairs or anything like that?
It definitely affects growth. I don't think I've noticed anything particularly with color, but that's actually a really good idea to start pursuing things that might affect color. I think I would love to get more into the breeding of them eventually. You don't see ornamental mushroom breeding really outside of psychedelic mushrooms. There's a lot of people doing that with psychedelics, but you'll see ornamental breeding also in things like reishi sometimes. There's some domestic growers who make really beautiful bonsai sort of sculptures with them, and they'll pursue things that make antlers more aggressive and the response to CO2 more aggressive. But by crossing spores, hyphae, and making strains that wouldn't necessarily exist in nature, I think there's value to that. And we experience mushrooms through science mostly and through their applications as medicine or materials. But it's very easy to just see them as something beautiful, and I think that will be the introduction a lot of people can have that will help sort of get rid of this fungophobia in society. But I think that's a really good idea, pursuing more brilliant and different colors. I've seen a lot of people pull out albino and variegated mushrooms, which have been really interesting to me. But I guess I didn't mention, I can preserve these. They are preserved when I first got into them. Dehydrated, ideally freeze-dried, put in a vacuum with beeswax and Damar varnish, which is a tree sap. That mix of wax and varnish has been used by the Greeks to preserve wooden sculptures that are still in perfect condition in museums now. So I have every confidence that that would be an archival process to preserve them that is sustainable and organic as well. But I've also experimented with epoxies and things like that. But all of these, pretty much all of them, the few I've sold to collectors, I didn't preserve. I told them, this is a conceptual art piece. It's going to degrade in your house. And they did. The best one, they come back to me at the next show I see them at, and they're like, oh, that piece, we went to Japan, and our housekeeper called us. And they said there was like beetles all over our house, and they'd gotten in and they'd laid eggs in the sculpture. And I was just like, I told you. I told you not to hang it. I told you it would put it in your garden or something. But yeah, I let them degrade. They become fertilizer. They become incredible fertilizer. The worms love them. And so that really closes the circle of the process, is that it's not a acrylic paint, or it's not a metallic sculpture that's going to sit around. It's the artwork's a process there. But it doesn't mean I'm not still looking into preserving them. Yeah.
You mentioned you had success inoculating guava. Have you tried other invasives such as Christmas berry or African tulip, and most importantly, the ones that are supposed to have antimicrobial properties like silver oak? That's really hard to break down just by itself.
Yeah, I've tried quite a few off the top of my head: guava, avocado, coffee, yerba mate, kukui. I have tried silver oak. There's a lot I'm forgetting, but Basically, no. I can get them to grow off that sort of stuff, but I will usually need to let the wood sit for a while, and the chemicals are probably leaching out that are problematic for the mushrooms. So I don't think that's helping as much, but I can get them to colonize and grow. I've probably been looking into the things that are easier, but I do think it's definitely a possibility. I think looking for different strains, adding a whole bunch of spores instead of a clone into something like that would probably give you more opportunity to find a mushroom or a strain that would like that. Okay. Yes.
So have you thought about making picture books like that Scottish guy makes, with his ephemeral pieces that only last a day or two?
No, but that sounds great. That sounds great.
He made that movie, Rivers and Tides, that everyone here will
Andrew Goldsworthy? Yeah, okay. Yes, he's a huge influence to me. His work,
He doesn't set out to make it.
yeah, so he'll go out into nature, he'll arrange some rocks or do something very poetic. I'm not doing justice by saying arranging rocks, but just his materials will be the things right around the forest. And, as soon as the rain comes or the river moves on, they'll be gone. And he takes a photo of them. And I think he was doing that in the 60s or the 50s even. He was doing that a long time ago. So now you'll go into the forest and you'll see people stack up rocks or something. It's very poetic, and he was able to succeed by doing documentation of that. But yeah, very, very influenced by him. I think his perspective gave people who don't go into the forest, which is a lot of art collectors, a lot of gallerists. Art's very centered in urban areas, almost exclusively centered in urban areas. And it's very hard to bring something into a gallery space. The showing living mushrooms in a space, people just want you to hang them on the wall, but it's gonna dry up in an air-conditioned room right away. So finding a way to bring the outdoor in and give people that experience, like Andrew Goldsworthy did, I think is really, really something we should all consider when we're setting out to be creative.
Are you a muralist too?
Muralist? No. But technically, I have the notes and resources now to be one. So if you know of any murals, I'd love to make a 40-foot mushroom panel. Sounds great. Yes.
Strawberry guava can be a little toughy. So have you had any trouble getting the mushrooms to actually take to that way?
Yeah. Yeah.
Species you've noticed?
The Pycnospora is by far the most aggressive. But yeah, the strawberry guava doesn't work well. And I do use it, but I usually need to let it leach out and probably break down by primary decomposers a little bit. Bacteria will break it down a bit before I sterilize it. Yes?
Do you have any experience experimenting with mycorrhizal mushrooms?
No, I don't. Specifically, like I said, my technology is pretty low. So, I very recently acquired a microscope, but hardly know how to use it. Really, it was what was growing. Just because I had such low technology, it was just what would outcompete stuff. And yeah, I would love to though. You got any cultures? Let me know. From Oahu. Yes.
How long does it take for those allelopathic compounds to leach out? How long do you let the primaries go for?
A while, 3 months.
Okay.
Yeah.
Do you ever intentionally use lately we had that oil spill and on our walk this morning they were saying that this particular species is good for breaking up petrochemicals. So do you ever take polluted soil or substrate on purpose because these are going to be non-edible, non-medicinal, so
I have not actively done that, but that I think would be a really interesting conceptual point to add to an exhibit or a process.
incorporate the microremediation
Yeah,
in the art.
absolutely. I think that's what I was considering with the coralline algae project, was representing a damaged environment, but I think that's a really great idea. Thank you. see you first.
Have you ever tried using multiple types on one panel?
On what? One what?
On one panel?
Yes, accidentally. Yeah, mislabeling grain and mixing it together. I think I did once have them coming into each other, and yeah, they do battle. They usually won't colonize all the way, but it is really cool. You see them come and meet and battle, and usually they'll just kind of stall out. But yeah, I had thought about doing a panel that was very literally one side meeting the other, and it'd be great to do that as a video, I think. I didn't show it, but I was very into time-lapse videos with the crystals back in the day, and I think it's been a little intimidating because technology has gotten to a point where so many people can do such good time lapses. It's hard to do that outside. You really need a closed environment to get the lights to stay stable, but the interaction, I think, is very poetic of them battling. But yeah, it was all accidental when I did it. Yes?
This is more of an idea, but the Museum of Modern Art in New York City has this beautiful enclosed outdoor sculpture garden with plants and stuff, and they have problems putting their usual art in that space because it's open to the
Yeah.
environment. But it would be perfect for something like
That's a great
art,
idea.
and they do short installations and stuff, so it's not going to be out there for years and years where it degrades. That would be
That sounds great. I'd love to do that. If you know any curators, please. That's another thing. Because of the fungophobia, people are very hesitant about moving these into spaces. Because a lot of galleries also house collections, and I'm fine with that. That's not my intent. But that botanical gardens in Hawaii usually have pretty elaborate greenhouses too, and stuff will fruit really readily in a greenhouse as long as there's pretty stable temperatures. These don't need like a full-on misting humidity system. Basically, if you just spray them down with a hose, and if you keep them submerged, these guys love to be underwater for a few days. They're just very, very versatile. Thank you guys so much. I really appreciate it.
Hi guys, my name's Kira. I'm a ecology, evolution, conservation biologist. I went to UH Hilo. I live on Hilo side still. And in my senior year, I started a project with Jeff Stallman, who's an amazing part of like the Hawaii mycology community. And yeah, I got into this project and we did 2 different papers that have been published now, and one of them was on seasonality, and then we did one on endemism as well. Today I'm mostly going to be focusing on the seasonality one, and then I'll touch on the endemism at the end of that. And yeah, thank you guys for being here. There's been amazing talks, and I'm really excited to be a part of this. So we focused on macrofungi and what are those and how are they classified. So we've got the phylum Basidiomycota, and within that is the class Agaricomycotina. And then today, and for the study, we focused just on like the Agaricales involved in that. And the Agaricomycotina are the Basidiomycota that form the macroscopic fruit bodies, and that includes like your mushrooms, jelly fungi, and some yeast. And then the Agaricales are the like fleshy fungi, those like gilled mushrooms, jelly fungi, gastroid fungi. And we did, for the focus of seasonality, we excluded those species that you can detect like more year-round, the ones that have like those persistent structures. So if you see like a conch on a tree, that's going to be there a lot longer. So we didn't include those ones and like what months are they found in. And those are like your polyporoids, your corticoids, gelatinous, and then the other species involved in that. And then what did we know kind of like going into this? So there was, in the past, there was like estimates on like the overall endemism of fungi in Hawaii to be like 80 to 88%. So endemic mushrooms, for those that don't know, it's the ones that are here and nowhere else in the world. They're like on native vegetation. And And then in our endemicity paper, we found it to be 83% is the number that we got. And then this is also, like pending on like, is the data that we have, like, is it accurate, like just based off of like morphological observations? Since we don't have like that much sequencing yet and like molecular data, it could change throughout time, and all of these like numbers and like everything can like evolve and like become more accurate over time if we're like all getting out there and like making all these observations. The more collections we have, like the more accurate our data is going to be. So like, please go out there, like survey, like Ben was saying, like iNaturalist, download it if you don't have it. I'm going to shout it out in one of the slides at the end as well. That's like a simple thing that you can just like, whenever you're just out on a hike, you can just like put in some data, and it's awesome. And then sequencing is going to be like super important. I'm going to like touch on that a bunch too. Some background on like the information we used. So we found 643 species of agaricomycetes that are known in Hawaii, and that's just from like stuff that's actually been published, like scientific papers and online sources that have been. But we also estimate that there's at least 450 species that remain undiscovered, and that's just like the mushroom-forming fungi. And there's like all the microscopic ones as well. So there's like so much more. So there's so much out there to be found. Like you can discover new species, and then so timing of surveys is going to be like vital for increasing detection, and that's why we wanted to like look at like, is there actually a mushroom season? Like, can we inform people like when are the best times to go out in like different environments, so that it can be more informed of like, so that your time is like more valuable. And we wanted to like collect a bunch of life history and phenology information to help people find mushrooms, and then like evaluate their conservation status. Is it like changing over time, and continue to like monitor these populations to see if with like climate change and stuff, like are they changing as well. And then so like places like the Pacific Northwest and other places like that, they have like this clear mushroom season, and it's like it correlates with like when like rainfall happens and when there's like these temperature variations. And like living in Hawaii, like it's a lot more just consistent, like year round. Like we don't have this like giant variation in rainfall and temperature. And so we were wondering, like, do we have like a similar type of thing where it's like we have this mushroom season? And that's what we were looking at. And so we investigated what are the best months to go out and look for like this increased Basidium mata, which is like the mushroom forming richness and abundance. So richness is going to be like your diversity, like how many species are present, and then abundance is how many like collections of all these species. And we like did a literature review, and then aggregated like the observation and collection data that are on these online databases, which I'll say which ones those were in a moment. And then we examined the seasonality based on average monthly temperature and rainfall. And separately, we did this for native species only and under different elevation and annual rainfall categories. I'm also going to touch on those. And then we hypothesized that Hawaiian agaricomycete species would show seasonal patterns in richness and abundance, and they would both increase in May, June, or July and remain elevated through January before declining in February, irrespective of average monthly rainfall and temperature, native or non-native origin, and annual rainfall and elevation categories. And we put together this species accumulation curve for species reports and observations in Hawaii over time. And people have been touching on this, like a lot of our observations are like way more recent and greatly increased over the past like 50 years, especially in the last 20. I think from like 1992 to 2007, we went from under 100 species known in Hawaii to over 600. And that's just continuing. Like our data collection that we did for this project ended in like May 2021. So there's like still like a lot that's happened since then as well. But and at this time, only 61 of those species had sequencing. And so, we have like a lot of work to do on that, which involves funding, which we had no funding on this project, so we didn't do any of that. But and only 3 of those 61 sequences were native species. And it'll be interesting to see if there's any major changes once we sequence more species. This could lead to realizations of species thought to be non-native as being natives. And this could open up the possibility of adaptive radiations in Hawaiian fungi, which is like a single lineage, like rapidly diversifying. So like in Hawaii Island, like we have like so many different like niches and environments. And so like one species could, or like one lineage could like rapidly evolve out into all these different niches and environments, and like it could diversify to fit that environment. And that could be a really cool possibility that hasn't really been looked into, and it hasn't been proven to exist here, but I think that it's a huge possibility of that existing. And then this is just like a preview of like the data sheet we did. We did like all this information for 643 species. This took me like a year and a half. And all the zeros correlate to no's, the ones are yeses. And the resources we used for this, so we did Mycobank to make sure that the species names were accurate and up to date. And we did change a bunch of the names, like from Dawn's list that was like in 2007. And then GenBank to see if the species have been sequenced, and then MycoPortal and scientific literature to determine locality, seasonality, and like the vegetative associations, if they're associated with native or non-native vegetation. And and then this is some of the data we collected, and these weren't included in our seasonality analyses, but this is like the fruiting body form was put into 6 different categories, and it's the major driver of diversification in mushroom-forming fungi. And the species were also designated as fleshy or polyporoid, and the polyporoids weren't included in this, which you can see at the bottom. Those are like the more persistent ones. And the lifestyle for each species was determined using Funguild, which is a database that was made by one of the labs at Manoa. And there was 4 designations. So we got like your saprotrophs, which are the wood rotting fungi, and the ones that are like involved in decomposition. And then symbiotrophs, which are including your ectomycorrhizal fungi that form these mutualistic associations with plants. And then pathotrophs are pathogenic, and then not available if there was like they were a combination of them or just we didn't know. And then lastly, in our data set, we looked at habitat type and external biogeography. And here you can see like the elevation categories that we used and the annual precipitation categories. So got your montane, lowland, subalpine, wet, mesic, dry. And then this map and this like external biogeography was used by Price and Wagner on like a study on angiosperms, and then we just adapted it to ours, and we added Eurasian because they didn't have European countries involved in that, and we included that category. And the majority of non-native agaricomycetes were in the widespread category, so they occurred in more than one of these regions. And then your highest number of species were in the wet and lowland habitats. Can anyone name any of these mushrooms that are up here right now? Yeah, we got hygroscopies. We got a couple of those. So we got hygroscopy Noelo Kalani in this one. And then we got hygroscopy Lama Lama Humidicutus Pelea. Humidicutus poilana. And then we've got Humidicutus, Pacella for this one. And then Pelea for that one. And then the bottom right is actually just, it's a Callistosporium species. We don't have an actual picture of Callistosporium venosobruneum, but that's what I wanted to put there. But and so these are 6 endemic Hawaiian mushrooms. and they are all categorized as like endangered, or they're vulnerable or endangered. So the IUCN, which is like the International Union for Conservation and Nature, they have a red list where they evaluate like threats to extinction, and these 6 have been evaluated. I think from seeing one of Ben's talks, I think there might be 2 more that have been evaluated. But only about like 425 out of millions of species on the planet of fungi have even been evaluated by this red list. And, you know, we have all these endemics here that haven't been evaluated yet. And I would love to see surveys carried out for these other endemics that aren't on here right now, and see if those species fall into these categories as well, so that we can monitor them, we can keep up with conservation of these. And And then, so this is one of the charts we made, and it displays the months that those species are found and that we've had collections during. And this is just 87 collections over like all of time. And some of them show strong seasonality. We've definitely got that peak in January for all of them combined. But there's some rareness in general, like Callistosporium venoso-bruneum, which we didn't have the picture of. Is only being found November and January. Humidicutes poilena was only in November. And then while other species, like Humidicutes pelea, occurs throughout more of the year. And these are our richness and abundance charts that we made. And the top left is going to be the total species diversity or richness, considering all the agaricomycetes that were in this study. And that's based off of 321 different species. And there's a peak in January, and then you see like the lower diversity from February to June. And then it increases in July and continues at an elevated rate through December. And then below that's just the native species richness, and that was 33 species that we included on that. And that's the February to April has that dip, and then June and October had low diversity as well, and then we see those peaks in like January and August. So those are good times to go out for those in the native forests. And then in the top right, we've got the overall abundance, and that's just based off of all the collections, and we had 3,285 collections involved in that. And there's that peak in January and in March and October to December. And then from the native abundance, huge peak in January, low February to April, and then kind of like lower continued out throughout the year, but a little higher. And then we've also got these graphs, and they're going to show the abundance and richness data on islands within annual rainfall and elevation categories by the average monthly rainfall and temperature. And the points, they're kind of hard to see. They're color-coded. We got the key down there. But that's like where the collections took place. And so we only included the categories where there was over 200 observations, so that we had like accurate data, like that it wasn't going to be insignificant due to like a low number. And then the black bars are going to show the abundance, and the gray bars are going to show the richness. That blue line is the monthly rainfall, and the yellow line is the temperature. And we saw that the Basidiomata abundance is positively correlated with average monthly rainfall in lowland mesic environments on Kauai and Oahu, and then lowland dry environments on Oahu. And so we can plan surveys in the future, like if you're going in these locations, they can be planned accordingly to this data and where the peaks occur. And there was not significant correlations between the Basidiomata abundance and average monthly rainfall or temperature in lowland wet environments on Hawaii Island and Oahu, and then in lowland mesic environments on Hawaii. And so in these wet areas, the like rainfall is a minimum of like 2-3 meters or millimeters, so not meters, millimeters a month. And and so like increasing rainfall above that baseline didn't lead to any increase because they're already like at that like minimum baseline to have like occurrence of like basidiomata production throughout the year. So to sum that up, like what is the mushroom season in Hawaii? Definitely highest in January throughout a lot of the data. Lowest in February to April, and then it increases May, June, July, remains elevated through December, and then we found like in the dry and subalpine locations, there's also a high diversity in March. And then there's definitely some like limitations to the data we had, and it's only going to get better. And there can like this can be like repeated by anyone. Like we had no funding, we were just like doing this. You just need time and motivation, I guess. But firstly, more collections. Like the more collections we have, the more accurate it's gonna be. The more we're gonna just understand like holistically. And then secondly, like there's a quote that the fungi are where the mycologists are, and we definitely see that bias. Like there's a lot more collections in Hawaii and Oahu. We're like in Hawaii Island, like we had Don and Dennis like do a lot of collections here. And then in Oahu, there's like a good aggregation of mycologists there now, which is awesome. But yeah, we need like more collections on Maui and Kauai and Molokai. And I know that people are doing that now, like, which is super cool. And then we also didn't consider like how seasonality of agaricomycetes may differ between substrates. So we got like wood versus soil substrates, and then between ecological roles. So we got saprotrophs and symbiotrophs, and these have been important variables in other places like a Japanese oak forest and like throughout Europe. And like we didn't look at this at all. So like that could be a possibility for somebody to look at. And then a lot more sequencing, which requires funding, but shout out to like the Hawaii Fungi Project. Like it's super cool to have these like sequences and like have more accurate data and know if like these species that we like thought that aren't native like actually could be native, or if they're like species that are on different islands like could be different from each other. And yeah. And then yeah, the data is biased more towards the recent past. A lot of the observations are just very recent, so like it'll be harder to look at like if climate change like is playing a part in like these like range distributions. And then like what's the conservation implications of knowing like what is the seasonality here? And so understanding this the seasonality of the production here could help managers to better plan out field efforts to monitor these populations or additional populations. And this is done a lot for native Hawaiian plants. I've worked a lot in conservation in Hawaii and like done this with plants a lot, and I would love to do this with fungi like way more. But and then all those like those 6 that I put on there that are on the IUCN Red List, those are all most often found in like Ohia dominant like native forests. And if you're from Hawaii, if you're not, like rot is a major problem, and it's like causing a lot of decline in these forests, and we have no information on like if the native fungal population is decreasing with that. And it'd be really good to like monitor these populations to see if they're like declining, or what is going on with that, if their range is declining. And yeah, so that's needed for the demographic and distribution changes. ROD is rapid ohia death. So it's, yeah, some pathogens that are killing off ohia trees, which are these native trees. So beautiful. And then, yeah, just like collecting this baseline seasonality data is going to allow us to look at like the range that we know of right now. And then in the future, we can like, when we keep collecting data throughout time, we can see like, are there a lot of changes, fluctuations with the changing climate, and generating like DNA barcodes for these threatened species to monitor for them, new population discovery, and then like taking environmental DNA studies, which is like you look at everything that's in like a soil sample and what's present in that environment. That'll help a lot with these comparisons. So future directions, where to next, sequencing, including this phenological data into the IUCN species threat assessment. So online you can go and like look at information about the species that are on like the IUCN red list, and you can like see their information, but we don't have this like phenological data on there. And I think that it would be really cool to include this kind of stuff in those assessments so people can get more information on them. And being able to record the absence of species as well as when are they present. It's also cool to see like when are they not present, and that'll be useful in gathering that range data as well, especially for the threatened species. And expanding our taxonomic scope, like beyond, like we just looked at like the agaricomycetes, and you can do this kind of thing for a bunch of different fungi. And then shout out to iNaturalist again. Download it if you don't have it. Get involved in this whenever you're just out hiking and walking about. And just some acknowledgements. Shout out again to Jeff. He's amazing and brought me onto this project, like advised me so much. And then Matt Knope was also my advisor on this. He's an amazing biologist at UH-Hilo, just great guy in general. And then Don, Dennis, for just paving the way for us to all like be here right now and like studying fungi in Hawaii. And everyone who put this on and everyone who's here right now, like mahalo nui loa. And then these are the papers we put out. I didn't get into like the endemicity as much in these slides, but I'll like touch on it real quick. So we also did a paper on endemicity. So we looked at so out of like the native species, what percentage of those are endemic, and then we compared that to there's like 5 other taxonomic groups in Hawaii, such as like angiosperms, bryophytes, that's like your mosses, and we looked at, so it wasn't the highest out of those taxonomic groups in Hawaii, but it was like on par with those as well. And then we also compared it to like 7 different groups of like oceanic islands and archipelagos, like what percentage they had from some collections that had taken place on those islands. And they definitely weren't as thorough in their collections, so that could improve a lot, but it was a lot higher. And so the theory of island biogeography that we're like referencing, that's so the more distant to a mainland that the islands are, and like the larger area leads to increased endemicity. And so we're the most isolated landmass in Hawaii, so we have like so many endemics. Like we have this huge like percentage of endemic species, and we found that it is similar in fungi. And yeah, so there's a lot of work that can be done on that as well. And there's like so many more endemics to probably find out here. And yeah, thank you guys again. Yeah.
If we've never used iNaturalist before, if there's any challenges that you've seen with like incomplete data being reported, is there like some best tips and best practices for using it?
I mean, like I've definitely seen some like really wrong, like people will just input like what they think the species is, or they'll just be like, "That's a plant," and I'm like, "That's a mushroom." But so I'm not exactly sure. I don't know if you have any answers to that.
Well, for your sakes, definitely take a lot of pictures, and obviously in mushrooms you want to do like the top and the bottom, you want to get as much data as possible. Obviously, like the more samples the better, and like life cycle shots, because mushrooms begin to look like other things if you don't, like a mature one versus a younger one. Like the more you can find, the better in different life cycle stages. But what's cool is it has a community element where there's like essentially fact checkers. You know, like there's experts that may not even live here. Like like Alan Rockefeller's, who's in California, is a mushroom expert, constantly like identifying things for us here. So luckily, there's a lot of fact checking, and I believe there is like research grade status of observations on there too. So like, yeah, I don't know exactly how who determines that and like within that? You guys.
As soon as three people agree on an identity, it becomes research grade. But yeah, really good pictures, and I was going to add something else. Maybe it'll come to me. You're going to add more.
Yeah.
I would add ecology is super important. So like taking pictures of the substrate it's growing on, or nearby trees, making like detailed notes, texture, smell, even taste. If you like chew off little pieces of mushroom and spit it out, you can do that.
Okay.
But pictures, a lot of pictures. You don't get overwhelmed with all the details.
The other piece I was going to add is that the desktop version is actually much better, much more like features and interface than the mobile version.
You can like mass upload photos instead of like one by one.
Yeah.
Chase, you?
On the topic of adaptive radiation, do you have species here in the archipelago and or genera that you're most interested in looking at that may be expressing what you think, hypothesize might be adaptive?
There was I wanted to do a project on, I forget what genera of fungi I was looking at, but with silver swords, I wanted to look at if there was like arbuscular mycorrhizal associations with those that may have allowed them to like have adaptive radiations, and like plants that have had all this like research on adaptive radiations and have been shown to have them already. I wanted to see if there was associations with those plants that maybe associated those adaptive radiations. But I don't know specifically which fungi I want to look at, but I would love to figure that out.
I'm just curious, what was the percentage of endemic species found on this island versus other islands? Because I know by far this island has more preserved native habitat.
Yeah.
Plantations and stuff, like lanais are totally wiped out, for instance, plantations.
Yeah, and we didn't even have like information for Lanai because there was like not really collections of fungi there. But we don't have that specifically, but I could look at it if you want to like exchange information. I can try to get that to you. I'm pretty sure, yeah, like Oahu and I want to say Kauai. I looked at my data in a little bit. Yeah.
On the book they say which islands they found it on.
Yeah.
So it's throughout the islands.
Thank you guys.
So as you probably know, mushrooms come in all shapes and sizes, some extremely large and some very small. That is my thumb in that picture in the right. This is not me, but that is my thumb on the right. Little baby mushroom I found in Ecuador. And they all, some glow in the dark, some are fuzzy, some have a crazy net. But they also can have some benefits for humans. And as weird as they are, fungi actually are closer related to us humans than we are to plants. And we have such a strong relationship to plants, but not as much with fungi. And there's even a theory called panspermia that fungi actually came from space and spores were hidden in an asteroid and landed on planet Earth way back in the day. And we can see how resilient fungi are by some of the examples of the extreme environments in which they live. So researchers off the coast of Japan were doing some underwater drilling underneath the ocean, and they discovered 69 different types of fungi living underneath the ocean. And they put some of the spores on a petri plate, and one of the species that they put on was a Schizophilum species. And not only did the spores germinate, mycelium grew, but it actually fruited a mushroom. And this was in hibernation for 20 million years. So it's like finding a 20 million year old apple tree seed, planting it. Not only does it grow an apple tree, but actual apples off that tree as well. Fungi have been found in Antarctica, in the snow and ice. They are thriving. The most radioactive place on the planet, Chernobyl. Fungi are not only living, but thriving there, and only eating radioactive isotopes as their food source. There's been examples of spores of fungi being found on the noses of rockets, re-entering our atmosphere There's a study that happened about 10 years ago where they took a piece of lichen and they threw it out of a space station. They let it float in space for 6 months. They brought it back on board, and it continued growing like nothing happened. So all of that to say is that fungi are incredibly resilient, and to be able to live and thrive in these areas, they must be producing a lot of crazy compounds, some of which are beneficial to humans. The oldest fungi ever found was about 1 billion years ago. The oldest, cap and stem mushroom is about 810 million years ago in the Congo. And then over the next 100 million years, we get this symbiosis on land, something called lichen, which is a symbiosis between a few different organisms, a couple different fungi, cyanobacteria, and probably, honestly, more. We're discovering more every single year with lichen. And so this was kind of the start of early land plants. And so these early land plants are covering this rocky surface of early earth and secreting enzymes into the rock, creating soil. And even before plants had roots, fungi actually acted as the roots of plants. And so this is an example of what maybe an early plant looked like. And the fungi would act as the root surface, the roots of the plants. And then mycorrhizae, or fungi that connect to the roots of plants, they didn't develop for another 30 million years. And then we get these things called prototaxites. They're huge kind of lichen mounds, and when researchers first found them, they're about 30 meters tall. They thought that it was a dinosaur bone, or maybe, they did cross sections, and they thought, kind of looks like a ring of a tree, right? And so they're like, oh, it's an early tree maybe, but it was about 90% fungi, give or take. And so this was kind of a giant lichen, so to speak. And some theorize that these were kind of early trees, or the start of early trees. And so as mycorrhizal fungi is developing around, 470 million years ago, give or take, and we start building soil, we start to create this mycelium, this internet of the forest. We start to see these great ecosystems happening beneath the soil. And around the time, we get arthropods coming out of the ocean onto land about 420 million years ago, and they start digging into the soil. And, they have a vast buffet of food. They have fungi. They have roots of plants that they can eat up. And so this new symbiosis between fungi and plants has to defend against this new threat. And so this is starting the age of chemical warfare with arthropods. And so it took about 300 million years before the first cap and stem mushroom that we know of to be found, to develop. And around the same time, we get cordyceps. So fungi starts attacking back against these insects and start actually colonizing the insects. And they start secreting these crazy compounds like ergotamine, one of the precursors to LSD. And this is Ophiocordyceps lloydii, which has a crazy, perfect rectangle-shaped spore. And we get various examples of, these entomopathogenic fungi secreting these really crazy compounds. Like this is Massospora, and some of the species, they secrete amphetamines into cicadas. Some actually secrete psilocybin into cicadas. This is Ophiocordyceps heteropoda, I think is how you pronounce it. And does anyone know, do these two compounds look familiar to anybody? Yeah. What mushroom are these in? Amanita muscaria. So this fungi is pumping the same, semi-psychedelic compounds into this cicada. And we get, instances of species jump again. So the same entomopathogenic fungi that were attacking the insects, they jumped hosts into fungi, and they started colonizing truffles and started, having a bunch of beta carbolines being secreted. And so we're seeing all these new chemicals showing up on planet Earth. And they're starting to kind of break away from their symbiosis. They're starting to go outside of their parents' house, so to speak. And so for hundreds of millions of years, they had this symbiosis with plants. And now they're starting to, explore into, colonizing insects and colonizing truffles and, saying, what compounds can I produce to survive in these weird new environments with these new weird food sources? And one of them is actually psilocybin. So, with the threat of insects, they're competing against the same food source. And so one theory of why the fungi even produced psilocybin in the first place, the magic compound in magic mushrooms, is it creates micro lesions in insects. So the theory is that, mushrooms were producing this as to ward against insects. And it has different effects on different insects, from suppressing appetite. So if a termite and a mushroom were competing against the same log, it can suppress the appetite of the termite so it can eat more of the log. And so we're getting all these crazy compounds from mushrooms as just competition against insects. And some of them are incredibly beneficial for humans, like psilocybin. We're getting all these amazing studies, can help depression, OCD, all these different things. But it first started as a defense against insects, or that's what the hypothesis is. And then we came along, unfortunately. Messed a few things up. And then the theory by Terence and Dennis McKenna called the stoned ape theory is that our early ancestors came along and we were following the dung of different animals, and we found psilocybin-containing mushrooms, and we ate them. And that was the precursor to our jump in evolution, our development of language, religion, etc. And I have a theory as well that at the same time, our early ancestors were eating insects that were pumped with these different psychoactive compounds from different fungi. And we're both eating the fruiting bodies of psilocybe species and these infected insects at the same time. But that's all theory, and it's a fun story. The first person that we have on record that has eaten mushrooms is the Red Lady of El Miron. And she had 2 spores of 2 different mushrooms wedged into her teeth. And so this is the first instance that we know of that a human was consuming mushrooms for food and potentially medicine. And then over the next, thousands of years, we get various cave paintings and works of art all around the world. This is the Tassili bee shaman in northern Algeria. We get petroglyphs in Spain around 4000 BC. Does anyone know the name of this mummy right here? Utsi, or Otzi the Iceman, however you want to pronounce it, was found with actually 3 different mushrooms, 2 on him and 1 inside of him. We get the birch polypore up here, which researchers think he was using to clear intestinal parasites. And then he was also carrying this tinder conch, and they think that he was using that to carry embers of a fire to start fires, but also using it as a form of moxibustion, which he was found with tattoos on specific meridian points on his body. And so he would smolder the mushroom to create heat under those specific points in the body, similar to acupuncture or acupressure, but with heat. And so using that to cure his form of ancient rheumatoid arthritis. And then he was also found with agarikon in his stomach, which he was eating for probably immune support. We get various mushroom heads and hieroglyphics in Egypt, which there's a lot of folklore around mushrooms in Egypt as well. We get petroglyphs in Siberia. We get these mushroom stones all around South America, Guatemala specifically. And then the first written text about mushrooms was actually from Hippocrates, who wrote about Fomitopsis officinalis, the agarikon, and he called it a cure-all. And then we get Bouveria bassiana. We get various texts in ancient Chinese medicine in the various materia medicas. They talk about Bouveria bassiana, reishi, porreococos, tremella. We get Ophiocordyceps senescens. And then it wasn't until 800 AD where the Chinese cultivated a red yeast, and it later turned into a statin drug. And so now we're seeing a ton of our pharmaceuticals are actually compounds derived from fungi, including ergotamine A, which is a really famous compound, penicillium, which saved hundreds of millions of people's lives, LSD, which is a fun one. We get cyclosporin A. This is if anyone has had an organ transplant or knows anyone with an organ transplant before, they've been prescribed cyclosporin A to suppress their immune system enough to accept a new organ. We get Taxol. We get a ton of different things coming out. And now they're kind of, here in the US, they're kind of hot right now. They're a new functional thing that people like to put in their smoothies. A bunch of celebrities are using them. A bunch of PR companies are talking about, it's this new hot thing, but it's been used for, tens of thousands of years all around the world. But they're definitely having a moment right now with functional mushrooms. And it's a lot of money as well. People are saying it's about a $6 billion market, sitting in the $90 billion supplement market, in the $650 billion functional food and beverage market. And they're growing about 811% depending on each functional mushroom. So good and bad thing, we're just getting a lot of people entering the space. Some people are great, like Malama mushrooms and a few people in the space, but other people not so good. My journey started in 2013, and this was my first week of college, which started with this red Solo cup, as maybe your first week of university or college started with. Except mine wasn't filled with beer. It was actually filled with dried psilocybin mushrooms. And long story short, I ate the whole thing on the spot because I didn't know what a proper dose was. And I later measured it out, and it was about 30 dried grams of psilocybin. Usually people start with like 2 or 3 grams as like a good starter dose. I took about 10 times that much, and just, I flushed my medication down the toilet the next morning. I stopped smoking cigarettes. I was smoking 2 packs a day before this experience and went cold turkey after. Was drinking, quit drinking, changed my friend group. And this was, wow, mushrooms can change your life. And I bought every book that I could. I signed up for everything that I could. I joined an internship at a functional mushroom company that was actually, they were just doing mycelium, learned some lab techniques, and learned about spawn production. This led me to Ecuador. I was studying leafcutter ants, which actually farm fungi. And they take these leaves and they eat them up, and they actually grow mycelium in their nests to eat as a sole food source. Until I found some insects with mushrooms popping out of their heads, and this totally changed my interest into entomopathogenic fungi and cordyceps. This is the first one I ever found, and if you were here yesterday, you saw a bunch more slideshows of the cordyceps horror show that I showed yesterday. But I also found an oil spill while I was there in Ecuador in Yasuni National Park. And it was kind of hidden. I was on a walk that I shouldn't have been in that area, long story short. And I found an unlined pit of oil. And I was reading at the time that fungi could have the potential to remediate these toxins. So I went home, changed my major, and I got some grant money, grew mushrooms on cigarette butts for a while, and wrote a book on microremediation. And this was kind of one way that mushrooms can be functional for healing. Because, we are one with the earth, so whether it's the earth or our bodies, it's kind of the same thing. And during this time, I actually got bit by a tick, and I got Lyme disease, and it just totally changed my life. And I had severe lethargy and brain fog and pain. I couldn't get out of bed. And I turned to cordyceps again that I was finding in Ecuador, and started growing Cordyceps militaris in my room as I was writing that book, as kind of my own supplement for my energy. And so for me, functional mushrooms kind of fall into three categories, and I really love this graphic. So usually when people think of functional mushrooms, they just think of the human body, right? They think of taking reishi to calm out, lion's mane for brain fog, things like that. But it can also be, microremediation, cleaning toxins in the earth and helping the earth heal, right? And can also be the mind or the spirit or the soul, and working with psilocybin and really healing that aspect, which kind of goes beyond the body. So kind of focusing on the body aspect, we're talking about Cordyceps militaris. This is one of my favorite functional mushrooms of all time, probably my favorite. And we had a big farm in Massachusetts for many, many years. And most people use this mushroom for athletic performance, for energy, help lung capacity. It's an aphrodisiac, so helping athletic performance in the bedroom and in the gym, and helping with if you're traveling, helping align your circadian rhythm, and all around just a really fun alien Cheeto mushroom. So in the wild, it actually grows on moth larvae. So it is an entomopathogenic fungi. It attacks insects. But for commercial scale, we grow it on supplemented rice. So most of the time it is vegan, and no bugs are harmed in the process. Then we got reishi. And so reishi is kind of an umbrella name for many different mushrooms in the Ganoderma genus, but the most famous being Ganoderma lingzhi. I'm probably butchering that name, from China. Also another famous one is Ganoderma lucidum from Europe. And we have various Ganodermas here in the US. I don't know what you guys have on this side or this group of islands, but this pictured is Ganoderma lingzhi, and what most of the research is about. And they keep changing the name, so a lot of the papers actually say Ganoderma lucidum, but it's actually Ganoderma lingzhi. And it's most famous for a group of triterpenes called ganoduric acids, and really amazing for seasonal changes, and calming down, really good for a good night's sleep. And really just kind of taking the edge off. It's one of the most popular adaptogens, so adapting to occasional stress and fatigue that we have. And also great for the cardiovascular system as well. Then we have lion's mane, Hericium erinaceus, which is kind of your brain's best friend. Kind of looks like a brain, or a white mane of a lion. And this, I love to cook this. You cut it in strips, and you do an egg and bread wash, and then it tastes like breaded fish. It's pretty crazy. Tastes delicious, but also really good in a tincture, powder, supplement. And it has a bunch of different compounds, which I'll cover in a little bit, that are great for nerve growth factor in the brain and cognitive performance, memory, etc. Chaga, not technically a mushroom. It's technically not even a sclerotia. It's like a canker or a conch. Half of this is like wood and hardened mycelium condensed together. And it takes most of its properties actually from the birch tree. And so if you grow this on any other substrate, it won't really be that potent. And so this is some of the problems that I'll get into of people producing mycelium on grain, is that they're not producing it on birch. And so you're not getting the properties. And so this kind of acts as like a semi-parasite, and the tree will try to send all these compounds to the infected site and the chaga will pretty much suck it up, so to speak. We got shiitake, and this is actually a scanning electron microscope of one of the gills, and you can see all the little pores inside, which is pretty cool. And I was growing this block in a tent with plants to cycle the oxygen and the CO2. But shiitake, really well-known compound in there is called lentinin, which is actually an approved cancer drug in some countries. But it's really great for supporting liver health and immune support. It's packed with 1,3,1,6 beta-glucans, which most functional mushrooms are packed with those compounds. We got maitake, Grifola frondosa, one of the tastiest mushrooms ever. If anyone has never had maitake, put it on your list. It's super good. I cook it with maple syrup and cinnamon, and it's like, oh my god, it's like a dessert. It's unreal. But it has a couple compounds in here that are famous 1,3,1,6 beta-glucans, which are amazing for immune support and also regulating blood sugar levels in already healthy ranges. We got turkey tail, which was actually on the cover of my book. That art is by Martin Bridge. He's an amazing artist that has some mushroom paintings as well. And probably the most famous compounds in here are PSK, and that is an approved cancer drug in Japan, actually, and has been used for decades, which is a 1,3,1,6 beta-glucan, which I'll say over and over again, great for supporting the immune system. We've got Poria cocos. This is technically a sclerotia. It's kind of like a yam, so it'll grow underground. And, inside, if you crack it open, it's pure white. And around the edge of the skin is kind of a pinkish color. And a lot of, depending on what part of this sclerotia that people use, some people use it as a diuretic to help detox. And it's also great for stabilizing mood. And so a lot of practitioners will use this as a mood support. And we're still trying to understand what compounds are in there to help support mood as well. Mishima, Felinus linteus, which is roughly translated into women's island, is great traditionally for overall women's health and grows on mulberry trees, particularly in Japan, but all around the world, and also great for supporting the immune system. We got tremella, one of my favorites. And it's actually a mycoparasite, and it's a weird mushroom. And this is if you feel it, it's super wet. I think we have a species here on the table. It's a jelly fungus, and so a lot of companies will actually put it in skincare because it holds up to 400 times its weight in water, and it's more effective than, I think it's called hydroloric acid, if I'm pronouncing that correctly. And so if you look at in the back of the bottle of many skincare products, they actually have this mushroom in there, surprisingly. And so amazing for supporting moisture in your skin. So is functional mushrooms legit? There's a lot of skeptics out there, and I understand, right? And just because it's been used for over 5,000 years doesn't mean it's legit. There's a lot of things that have been used for thousands of years that are not. So just because it's been used for a long time doesn't mean it's legit. However, there's over 600 human clinical trial papers published, and over 40 of which are on the NIH website. You can look these up, and some of the studies have up to 68,000 participants in one single study. So it's not like 10 people, it's 68,000 people in some of these studies, and there's estimated over 130 functional benefits. Now, 4 things to keep in mind. If you're looking for a good supplement or a good functional mushroom, I like to check off these 4 things, which get a little bit complicated, but this is kind of the good benchmarks to go through. So number 1 is you got to get good mushrooms. The first part of that is making sure they're actually mushrooms. Now, this thing started in the United States that's really weird, but a couple, producers figured out that they could just grow the mycelium or the roots of the mushroom and just slap on mushrooms on the packaging and kind of skip out on a lot of the cultivation, the time, the money, and there's no mushrooms in the products. That's only really a United States phenomena. You don't really see that anywhere else. But make sure there's actual mushrooms in the stuff that you're using. And then this gets a little bit complicated, but it depends on the cultivar or the strain, the substrate, the pH, the temperature, how it's dried, how it's extracted, and there's which all go into the potency. We're not exactly there yet, so for now, just make sure they're mushrooms. The second is make sure there's good extraction. This can get also a little bit more complicated, but something to go home with is most mushrooms have polar and non-polar compounds. So water soluble or oil/alcohol soluble compounds. So make sure it's dual extracted. And so make sure they're using both water and either alcohol or oil or another non-polar solvent. Number 3 is good certifications. Not every certification is perfect, right? But it helps to have USDA certified organic. It helps to have non-GMO. It helps to be NSF CGMP compliant, which means you're following, rigorous food safety and FDA compliance. You're not just making it in, your dirty kitchen. You just picked your nose and you're just like stuffing your hand in the powder. It's making sure you're following clean protocol. Then number 4, also a bit complicated, but good dose. And this will actually is dependent on these top 3, because you can get a pretty poorly grown mushroom or mycelium that can be really weak. And then you can have a really high quality variety. And so a gram of the really poor quality stuff is not the same as a gram of the really high quality stuff. And so you think of, if anyone smokes weed, weed 40 years ago is way different from something you can get at a dispensary now. So a gram is not the same as a gram. And there's something that I keep hearing people are like, I need a gram of functional mushrooms a day. I don't know where that came from, but it doesn't mean anything. And so this is kind of a weird, I can't really give you a good answer. There's no specific dose that you need per day. Every single person is different. Their body is, some people are more sensitive than other people. And there's varying different strengths of the product. But just making sure it's good mushrooms, is good extracted. It's extracted well, and then there's good certifications. I think you'll find an amount that works for your body.
So many of the products are made from parts grown in China, and they claim to be certified organic. How does that really work? How do we really know that they're really certified organic?
The USDA is a US-based program, and so it doesn't matter where it's originally grown. There's certifying agents that go to any country and go through the farm and making sure they're following the USDA certified organic standards. And so it doesn't matter the country of origin. It just matters the standards that the USDA fills out. But you'll have certifying agents going to the farm, working with them to making sure they're following the protocol. Same as a farm in Hawaii or Oregon or whatever. They're all following the same rules. So to understanding the first part of good mushrooms, it's important to understand kind of the life cycle of a basic Basidiomycota. Every single mushroom is different. This is kind of a general life cycle that might help. But they start as mushrooms, cap and stem, shoot out some spores. The spores come together to make hyphae and eventually mycelium to complete the cycle again. Now, in cultivation, we're going to talk about this later, but there's something called spawn. And so you'll grow mycelium or the roots of the mushroom on grain, either oats or rye or sometimes rice or sorghum, barley, whatever grain that you're working with. And you'll use this as kind of seed, so to speak. And some producers, in the functional mushroom industry were like, why don't we just stop here and grind it into a powder and put it into a capsule and call it mushrooms? And so most of the stuff that you're seeing on the shelf, unfortunately, is just this. It's just it's just tempeh, basically, ground into a powder. And it has a significant fraction of the functional benefits that you're looking for. And sometimes they don't even extract it, so the compounds are not bioavailable to your body. And it's up to 90% grain. Now, there's a way that you can grow pure mycelium in a bioreactor, and that is an option that a lot of countries do, including Japan and China and a few other countries. But still, even if it's pure mycelium without the 90% filler, it still has a fraction of the functional benefits. And this is actually against FDA guidelines to sell something that contains mycelium and label it mushrooms. The FDA says this is very confusing for consumers, and it is against FDA policy to do this. However, a lot of companies still do it, and they're still making like $70 million a year, and they're going kind of unpunished, which is, it's just a matter of time.
Yeah, so Paul Stamets is one of these people that sells a product that's only mycelium. He says he's hired many scientists to test the efficacy, and he swears that there's a lot of science that the mycelium is producing the chemicals and enzymes that are antimicrobial because the mycelium is in the interface with the bacteria and other fungi. And so Paul Stamets says that he's got whole groups of scientists testing the mycelium for its functionality. And Paul Stamets, he swears by his product, and I'm sure I've taken it and felt the benefit, and I'm guessing plenty of you have taken Myco-Immunity or Stamets 7, and that's one of these products that, maybe isn't, the FDA says is mislabeled or something, but like I'm just saying, Paul Stamets, he stands by his product, saying that his mycelium extracts are functional.
How does he label that product?
He changed it to mycelium. It used to be mushrooms, but he had to change it. I'll go into that specifically, but there's 0 scientific papers that show that the mycelium has more functional compounds, and there's dozens, if not hundreds, that show the exact opposite of what he's saying. There's nothing to back what he's saying. Actually, there's hundreds of papers that show that what he's saying is absolutely not true, which I'll go into. So you'll see things like this, certified 100% organic mushroom powder. These are from companies that, once you look at the other ingredients or things like that, you'll see mycelial biomass and fruiting body. And some companies will even go to say full spectrum, right? If anyone was in the cannabis industry, that's a great word. Full spectrum means like, oh yeah, we got all the benefits in there. And so, a lot of these companies will say, oh, we got the primordia, we got the spores, we got the enzymes, we got the substrate, we got all of it. It's the full life cycle, full spectrum, it's amazing, blah, blah, blah. But really, all they're having in there, they don't actually have full fruiting bodies. They have something called primordia. And so if you look in this bag, these little dots on top, that's primordia, and that's what they're calling a mushroom. It's probably like not even 1% of the whole product, and it's still 90% filler. And again, I'm going to go through all the studies that show, why that's bad. So that's like going into a dispensary, and they hand you a bag with dirt and roots and stems and seeds and a little bit of nug, and they're like, dude, full spectrum, man. Like, this is super potent. It has everything in there. And it's like, no, we know that what you want is the nug. And I don't want the dirt in there. I don't want the stems, let's be real.
I'll just say I've gone into debates with other Chinese medicine practitioners before on what he said, and I'm so pro fruiting body, and some people are like, oh, well, Paul Stamets over that, and that, and it just really makes sense when you think about the bigger picture that fruiting body was always used.
And I do want to say I have a functional mushroom company that uses 100% fruiting bodies. And I say that by saying I'm biased, but it's way more expensive to use fruiting bodies. Like, I actually would love for the research to show mycelium because I would save so much money. Like, as a pure capitalist hat on, like, I would love the research to show mycelium, but unfortunately it doesn't, and I wouldn't be able to sleep at night if I was giving people a product that I knew was like a 30th of the functionality. So like I am actually rooting for mycelium, but the studies are just not showing it. So
Yeah, thank you for all that. I remember years and years ago reading vaguely that like the PSPs and PSK, polysaccharide peptides and crestin in turkey tail, the way that they do it in Japan is do those bioreactors, and that these are actually secondary metabolites that are being secreted by the mycelia, as in nature when they're going through the soil, they're antifungal, antiviral, antimicrobial, so that they can cruise through the soil and eat their substrate, and then they harvest the secondary metabolites for the PSP and PSK for their cancer medicines. And it's interesting that we have this big debate right now about the mycelium versus the fruiting body, but in the case of PSP and PSK in particular, I hardly hear anybody talking about secondary metabolites and bioreactors. And I'm curious your thoughts on that, and Martin Powell's work in the human clinical trials, and the advantages of using both.
with PSP and PSK, it's for drug isolation, and so they need to use a bioreactor. They need to use mycelium to extract it because it's chemically pure and sterile. So it wouldn't be. Yeah, it wouldn't be sterile if they were growing the full fruiting body. If for drug development, they use bioreactors with that. But PSP and PSK are in the fruiting body as well.
Okay.
Yeah.
Cool. Thank you.
And a lot of companies, like I said, don't even extract. They'll just hammer mill or they'll freeze dry just the mycelial biomass, blend it up into a powder, call it a day. There's a famous study by Bastyr which shows that just doing a hot water extract, which is, half the extraction that you really need, significantly boosted the immune support that people had. I had to cut out some of the top of the study because FDA compliance, but basically it dramatically increased immune support by just doing a hot water extract. So this is a simple test that you can do at home if you don't really know if they're using mycelium or a mushroom. In this test, I used to actually blank out the names, but, whatever. This is our cordyceps. This is host defense. This is ohm. And this is just rice powder. And what you can do is you can drop iodine into these flasks, and it will react with starch and turn the liquid a dark purple to almost black color if it has fillers in the media. So let's see what happens. These are some of the most famous mushroom companies in the world, and they're just filled with fillers. Almost the same color as just pure rice. And we partner with Bastyr University. This is Reishi Strauss, who is spearheading the project. And we partner with her for a couple years, just researching with our cordyceps and a couple different growers on what was the difference between cordycepin and adenosine, the two major compounds in Cordyceps militaris, in the fruiting bodies compared to the mycelium. And we found year over year that the fruiting bodies had up to 51.5% or times more cordycepin and about 7 times more adenosine than the mycelium/mycelium on grain. And then if you're curious on the best extraction protocols, you can snap a picture if you want, or I can share the slide after.
50 times more, not 50%.
50 times more, yeah. Way more.
So if I saute some lion's mane in butter, would it be considered a hot water extraction as well as an oil extraction?
Technically, yeah.
But jokes aside, do you lose a lot of it if you cook the fresh mushroom versus making it into a tincture?
Yeah, you're still gonna get a lot of benefits, and there is certain there's just a joy of eating mushrooms that I just, I'm never going to tell people, like, don't eat mushrooms. Like, eat lion's mane. Yeah, it tastes really good. Go for it.
Was that comparison with mycelium versus fruiting body, or fruiting body versus mycelium on grain? And were they fruited or unfruited mycelium?
Mycelium on grain, and they were unfruited. Yeah. Which is the common practice of you don't fruit it and then use that. There's this other study out of China that showed that fruiting bodies had higher levels of cordycepin and pentostatin than fruiting bodies actually on bugs, which is pretty cool, and also pure mycelium in a bioreactor. And this had about 7 times more. It doesn't show it, I guess. Maybe it cut out. We sent some again, I crossed these out, but I'll tell you what they are. We sent some samples. We just pulled them off the grocery store shelves. We sent some samples of cordyceps into this lab out of Ireland called Megazyme. They test for 1,3,1,6 beta-glucans. They're the world's experts in those compounds. And we found that the fruiting bodies of cordyceps had up to 400 times more 1,3,1,6 beta-glucans than mycelium on grain. And so you can see the percent beta-glucan. This is, actually, I can't remember what they are, so I won't tell you. But these are a Whole Foods, one, and then host defense. So up to 400 times more in the fruiting body. There's other studies that show they have up to 13 times more amino acids. We see that in this one study that, a certain cordyceps mycelium sample that was taken had about the same 1,3,1,6 beta-glucans than just sorghum. So it's about the same as just grain. So another misconception that these companies will say is they'll put on their label, we have this many milligrams of polysaccharides. We're packed with polysaccharides. All that means is just long sugar chain. And starch rice contains about 74% starch, so they're packed with these polysaccharides. This to me is saying, hey, we have a bunch of fillers in our product. And we're just putting a big word that makes people starry-eyed and makes people think that it's good. But this just means fillers to me. Now, 1,3,1,6 beta-glucans is a polysaccharide, but it's specific. And there are even, I've started seeing companies just say generically, oh, we're packed with beta-glucans, but it depends what beta-glucan you're talking about. The one in fungi that supports the immune system is 1,3,1,6 beta-glucans. The one in cereal grains and starchy material is 1,3,1,4. And so there's a big difference. If they don't specify, that's a red flag. And I've also seen companies claim that they're packed with this compound, which is actually a dietary fiber found in oats. So it has nothing to do with fungi, and you're better off just eating oatmeal. It's a lot cheaper too. So moving on to reishi, it has up to 28 times more 1,3,1,6 beta-glucans, and there are many studies showing it's completely devoid, the mycelium, of the most famous compounds in reishi, which are the ganoduric acids A through D. There is no ganoduric acids A through D in the mycelium, which is literally the most famous compounds in reishi, which you'll have that bitter tasting compound, which is amazing for a natural inflammatory response in the body, post-workout, or other things like that. There's about 16 times more ergosterol in the fruiting bodies of reishi. And there's a study published that showed that up to 70% of all reishi products on the shelves today don't actually contain any reishi mushrooms at all. So it's really bad. I mean, it's really, really bad. And so most people are just, they're picking up, it says mushrooms all over it, and there could be no mushrooms whatsoever. and sometimes not even mycelium. So I have to play the devil's advocate, and I kind of want to because I am, in a way, rooting for mycelium to save me some money in my company. But, so there are 2 compounds in the mycelium of reishi that are actually higher in the than the fruiting body, and they're GABA and lovastatin. However, the recommended daily dose of GABA is about 100 to 300 milligrams per day, and the amount of GABA in the mycelium is about 100 milligrams per kilo. So you need a kilo of 1,000 grams of pure reishi mycelium to even get the daily recommended dose of GABA. So not going to happen. And lovastatin, again, you need a huge amount of the mycelium to even get the daily recommended dose. And there's up to 800 scientific papers showing that it could be potentially harmful for human health. So, again, yeah, these 2 are higher, but does it really mean anything? Not really. Maitake has these 3 compounds that are not present in the mycelium, great for vital protein health, cell health, signaling, and much higher levels of trehulose, nucleotides, MD fraction, which is the most famous 1316 beta-glucan, and up to 9 times total 1316 beta-glucans in the fruiting body of maitake. Shiitake, again, up to 4 times more 1316 beta-glucans, more amino acids, protein, glycogen, etc., much higher amounts in the fruiting body. Now, chaga, again, not a mushroom, but there's a difference between the sterile conch and mycelium on grain that some people are producing. Again, like I was saying, most of the compounds come from birch, and so there's various studies that show literally dozens of the compounds that are most famous in chaga do not show up in chaga mycelium that is sterilely produced. And so you get betulin, betulinic acid, melanin, lupiol, etc. do not show up in the mycelium at all. So that should be frightening that, why are you even taking it?
Is that even when grown on birch? Do you know anyone experimenting with that?
I would assume that it would still follow the same trajectory of, it would have a fraction of the amount. But most producers are just growing it on rice or oats.
Is there a difference between the fruit body grown on rice or the fruit body grown on a birch tree?
Of chaga?
Yeah.
No one uses the fruit body of chaga because it's but the sterile conch, you mean?
Can you produce a sterile conch on a grain block?
No.
No.
No. There's some researchers in Finland working on it, but they're still in the beginning stages of growing it outdoors and inoculating birch.
Maybe growing it on like birch sawdust would be.
Yeah, I think you need a living tree.
Yeah, and as you said earlier, it's a parasite, so that living tree is sending all of those all that stuff into the conch. Whereas when we're growing it artificially, we're not going and infecting living birch trees, right?
Yeah. But still a mystery to crack is cultivating chaga. Wolfiporia cocos, has up to 6 times higher triterpene acids in the sclerotia compared to the mycelium grown on grain. Now, lion's mane is one that has a lot of controversy. So, there's a couple studies about aranesine A being really great for nerve growth factor, and it's amazing. None of which are using human clinical trials. So every single human clinical trial with lion's mane is using 100% fruiting bodies. So every single human data that we have is using actual mushrooms, and it has about 16 compounds in it that are not in the mycelium that have been proven to support nerve growth factor. And this aranesine A that they're using for this study is a genetically modified organism to help produce more of that aranesine A in a pure bioreactor. And that's not what the companies are using. They're just growing mycelium on rice, and they're not using that that genetically modified organism.
I don't use Paul Stomach's product anymore, but I remember that lion's mane was the only one back then that labeled actually and fruit bodies.
Yeah, like I said before, most companies that label and fruiting body or full spectrum, it's just primordia. So it's just these little things, but even still, even if they use the full fruiting body, you're still getting up to 90% filler. And even if you're using pure mycelium and the fruiting body, you're still diluting the rest of the compounds.
Must have been aware of the lion's mane specifically. So.
Yeah. And so it has, the fruiting bodies, again, 29 times more 1316 beta glucans. And we looked into using, we contacted bioreactor specialists to produce just arenacine A. And we realized we can't do it organically. We have to use a genetically modified organism. They have to pump it with a bunch of chemicals. I mean, it's a really gnarly process what they were using for that specific study. And so, with just mycelium on grain, you're not getting a significant amount of that compound, which is why they had to do that.
If this is illegal, according to the FDA, why haven't we found an attorney and gone after a class action lawsuit? Because it's FDA.
I'm shocked that there's not a class action lawsuit. Like, I'm seriously shocked that there's not. I'm just kind of waiting around. I won't be that person that does it. But yeah, I feel like the more people that transition out of cannabis, and they sold their company for $400 million, they're super cutthroat, and they want to do that. I feel like it's a matter of time before that happens. And I don't want people to, I never want to have anyone hurt or lose money or whatever. I just
Responsible for their actions.
I just want people to say the truth, and that's I want people to be integral, and that's all I care about. I don't want to hurt people ever. I just want to, if you're doing something, just tell the truth. And then that's it. So we have a long way to go in the US. And I talked about this a little bit yesterday when I was looking at some various farms, and they had, all these different cultivars of Cordyceps militaris, and one had more iron, and one was packed more with cordycepin, and one was higher in adenosine, one was better in a tincture, one was better in a tea, it was a lot sweeter. And so we have a long way to go when it comes to specific cultivars. Like cannabis, you go into a dispensary, they're like, oh, this is Maui Wowie bubble gum, and this is if you're this, this is, you'll have a crazy art project, and you'll chill on the couch and want pizza, and so we have a long way to go with mushrooms, and I think we'll get there first with psilocybe cubensis and then functional mushrooms as well. And we're actually the first company to have a QR code on our packaging to show the lab results. And this is something that I want to see a lot more companies do is, have a standard analytical fingerprint of what's inside, right? So we can do that with cordyceps. We can say, hey, this one, and I'm starting to see it with cubensis is hey, this one has higher levels of baeocystin. This one has higher levels of aruginacine, which actually might be better for someone with Parkinson's, right? And so you can match the actual fingerprint with your specific needs. Another thing is it the mushroom you say it is? Chaga, for example, I've seen tons of people pick a canker on a tree, and they say, oh, this is chaga. And I'm like, that's not even a fungus. That's a canker. Or they'll pick, something, some other polypore, and they're like, yeah, this is chaga, right? And it's like, no, it's not even on birch tree. Come on. And so it's not even that, but also using, Ganoderma, Aplanatum, or some other kind of species of Ganoderma that there's no research on. And that can be a little tricky of, just because there's a bunch of papers published about the benefits of this species doesn't mean it translates to that one. And the same with cordyceps, the same with lion's mane. There's some companies using weird varieties of Hericium and just saying it has all the same benefits of Erinaceus. So, making sure it's the mushroom you say it is. So myth number one, mycelium is better than fruiting bodies. Unfortunately, 99% of the time, no. And you're better off with just 100% mushrooms. Full spectrum, we went over kind of bullshit. Sorry, there's kids. But full spectrum, you just want to go with the 100% fruiting bodies. There's some companies that say you can't separate the mycelium from the grain, and you need the grain is better. No, you can grow it in a bioreactor, pure mycelium, but it's still weaker than 100% fruiting bodies. If you see polysaccharides or just beta-glucans and not specifying 1316, stay away. That's a red flag. Paul Stamets went on a podcast a couple years ago and said Megazyme test does not work because it literally proves that fruiting bodies are way more potent. This is just not true at all. This is Barry McCleary. He's been studying 1316 beta-glucans for the last 40, 50 years. And he's a world expert on this process. And we brought him onto the podcast to literally get his opinion on what he said. And he said, yeah, he's not an expert, is the basics of it. But you can listen to the episode. It's a really good one. And then I was talking about this before. 1 gram a day keeps the doctor away. I've met so many people that say, like, you need a gram of mushrooms a day. I don't know where that started. If you look at papers, they range from like 0.1 milligrams or 0.1 grams to up to like 7 grams a day are some of these clinical trials. I don't know where 1 gram came from, but it depends on all these different dosages and things like that. But the future of functional mushrooms in the U.S. is really, mimicking a lot of the mushroom culture that we see in China, in Japan, that are really like thousands of years ahead of where we are. And the scale and the technology that they have in these countries is immense. And we have a lot to learn in our mushroom culture. So, this is a company that I saw that literally they're like, we will buy the land, we will build your building, we will do everything from like plumbing to electricity to security. We'll hire all your employees. We will set up all this automation equipment. We will design a business plan. We will like literally lay out a whole, like everything. And it's fully automated, and they're producing thousands and thousands and thousands of pounds a day of any mushroom that you can imagine. This is a cordyceps campus that I went to, and there's a cordyceps university. There's a cordyceps museum. There's cordyceps. There's cultivation zones. I went to a cordyceps cafe where they have a bunch of cordyceps dishes. I mean, we're way behind here in the U.S. And also our practices, right? Like if we were really following good laboratory, agricultural, manufacturing, production, clinical practices, we wouldn't have mycelium on grain. We wouldn't have these things to begin with. So as we're producing, as this gets more popular, let's have some good practices. And new extraction techniques as well. Supercritical CO2, ultrasonic. There's all these different ways to work with making sure each gram of mushrooms, we go through all this effort to grow it, making sure that we're getting every ounce of benefit from them. And also to find new species. We're like really working with like 10 here in the US. And if maybe 3 are the most popular. But if you go other places in the world, they're using so many more species that we just don't really use or know about. To put it in perspective, we have about 5 million estimated undescribed species of fungi, and about 120,000 of them we've described, and only 14,000 are actual mushrooms. Q. fungarium that we talked a couple times this fest about. They're the biggest fungarium in the world in England, and they take specimens from every single country, preserve them, so we can do this analysis that's really needed. And this is a mushroom report that they do every 2 years, and this is a distribution of all the new species found. And it's really weird, like 4% was found in Africa, only 12% in South America. And this has the whole Congo. This has the Amazon rainforest. Like, this is not a map of biodiversity. This is a map of resources and, experts on mycology. And so we really need a redistribution of resources to find new species and to use them to solve some of our world's biggest problems. Now, if you go back to this map, and then this map, this is just the land masses. We're missing 70% of our world, which is the ocean. Out of 326,000 natural products, 30,000 come from marine life. And over 1,000 compounds from marine fungi are discovered every single year. And in 1988, we found various compounds from marine fungi to treat H1N1 flu virus and HIV, herpes, staph, various cancers. And even for microremediation, the ocean is a wealth of biodiversity that we're just not talking about.
What's marine fungi?
Fungi in the ocean.
Like, how can they live without oxygen? I thought that they needed oxygen to breathe.
I don't know. I have no idea. I don't know anything about marine fungi. That's my point, is that no one does. Like, I have no idea. This is so cool. Like, no one's doing this. Like, and it's 70% of our planet. I have no idea how fungi are down there.
Oxygen is present in ocean water.
Chytridiomycota in the ocean.
So, beyond just storing these fungi, finding them, storing them in dusty cabinets, we need to do the research to figure out what are the compounds in there. So we get this Korean mushroom culture collection. We get the State Key Laboratory in China. We get API in Canada. They're actually utilizing some of these fungariums to test what compounds are in these new fungi. And to put it in perspective, we're finding so many more with advanced molecular biology, with rapid DNA testing that's getting cheaper and cheaper. We're finding new species that this is just on cordyceps. We're the second country with the highest amount of cordyceps species. So, I was looking this morning, I didn't find any, but, please look. Not enough people are looking for these weird fungi like entomopathogenic fungi or marine fungi or, fungi that live in leaves and weird places that people aren't looking, right? They're looking for the fleshy, the big mushrooms that jump out at you, but not the weird ones that can have some of these really interesting compounds. So Cordyceps guandangensis is an example. This was found in China in 2005. It was named in 2008. We figured out how to cultivate it in 2009 and 10. They did composition analysis, figuring out, the compounds inside. It was described as a novel food in 2013, did clinical trials, and from 2014 to present, it's one of the most popular functional mushrooms in China right now. And it was only named in 2008. So we could find a new species at this festival today, and in 15 years, it could be one of the most popular functional mushrooms in the US. So really, we have a world to discover. Asteria sinclairi has this compound myriosin, which they made into a drug, figoliamide, I think is how you pronounce it, but they use it to treat multiple sclerosis, and it gained $2.5 billion in revenue in 2017. This weird fungus growing on a cicada. If anyone found that, they would be like, ugh, this is gross, and just pass it by. But some of the weirdest fungi can have a $2.5 billion revenue and treat multiple sclerosis. So look for the weird ones. And also the enzymes as well, the secondary metabolites. This is Novozymes. Please, people, look it up. They're one of the biggest fungal companies in the world. And when you put on jeans or leather, or you write on a piece of paper, the next time you do laundry, know that fungal enzymes were extremely important in that process. And next time you fill up your car with gas, that ethanol was made with the help of fungal enzymes. And so our world would fall apart without fungal enzymes, and most of that goes unnoticed. This is Juliana Ferchi, and she's a really famous Chilean mycologist responsible for the Fungi Foundation. And now they're expanding globally and working to conserve fungi all around the world, which is important if we're going to find these new species to work with. So when I was at the medicinal mushroom conference in Nantong, China in 2019, this guy comes up and he says, I'm hiring so many people. When I hire them, they don't really know much about mycology. So we have to train them for many years. And, he's like, there's got to be a better way. So I made a whole university from preschool to PhD to train people on mycology. And so we need that in the US. We have no education system for fungi. And, it's up to us in these groups to share information. And we need that. We need more of that. We need more books being written, more documentaries being filmed, more museums, more places where we can share cultural and historic information. There's actually a cordyceps museum in China that I went to, and this lovely fellow that I'm sitting next to is Yushui Tan, who has grown the most amount of cordyceps out of anyone in the world, and is extremely humble and one of my favorite humans on planet Earth. But you can do really cool things besides, ingest these functional mushrooms. I'm actually working on a project right now, which I can't say too much before it's released, but we're using a specific functional mushroom in a part of a battery. And it's a fungal battery that can now turn on light bulbs. And it's working better than commercial batteries that are in the store right now. We can also have mycomaterials, right? We're talking about mycoremediation a lot, but a lot of functional mushrooms are being utilized to, create new styrofoams, to really replace the problem before they start. And so, it's amazing that we can clean up plastic and oil spills potentially, but, replacing the problem before they start. And it's becoming pretty popular with Lululemon and a bunch of really high fashion people as well. Mycroma, I believe they're from Argentina. They're replacing yellow 5 and these really toxic dyes with fungal dyes that are really a lot better for human health. And some of them are they come from these functional mushrooms as well. So if we can replace these really toxic chemicals in our food with things from fungi, I think the more the merrier. Another example of that is a bitter blocker from Ophiocordyceps sinensis, a really famous functional mushroom. There's a compound in there that actually blocks the bitter receptors on your tongue, and so we can use less sugar. Sugar acts in the brain the same way as cocaine. We're pretty much all addicted to it in this world nowadays, and so if we can use less sugar in our foods, I think the better. And also, there's research on using fungal sunscreens. There's a big debate on are sunscreens even more toxic than UV radiation itself? Do they cause more cancer than the sun itself? And a big debate now. There's a researcher using fungi from Chernobyl to make sunscreen for astronauts and even people on Earth. The same fungi that eat radioactive isotopes, they're using to create a cream to protect us from the sun. Now, I cover a lot of these different topics. I have a podcast, the Mushroom Revival Podcast, if you want to tune in deeper. A little product plug. We have, capsules and gummies and all that stuff if you want to check it out. But check out Malama Mushrooms as well. They got the goods right here. Buy local, buy from them. They're awesome people. And then also self-plug. I got a book coming out May 2nd. So coming out soon, guys. Check it out. I think we got a copy floating around if you want to peek through. Yep, that one right there. And this is a group, this is a women's co-op in Rwanda that we work with. We plant about 10,000 trees a year all around the world and donate a bunch of product to BIPOC communities around the U.S. during COVID and beyond. But it goes beyond just, functional products. And we're really psyched about trees too. Plants are cool too. And so it goes back to these three concepts, right? You know, it's like amazing to take functional mushrooms for your body, but let's care for the earth as well. And, you know, the soul beyond that. And let's eat more psychedelic mushrooms and become better people. So much love. Thank you. Do I have time for questions? Yeah.
Well, first, as someone that gives this talk a lot, that was awesome. Thank you. Second, how dare you disgrace our leader, Paul Stamets? What is your best guess as to why because that's someone that got into mycology, probably similar to you from Paul. Why do you think he's on his MOG, mycelium on grain, kick?
So I want to say he's still one of my big inspirations, and I think he's an amazing educator and influencer. But this is a topic that goes way beyond mushrooms, is like, don't get your information from influencers. Just because they have a lot of, social media followers or views on YouTube doesn't mean they're right. And I would trust, more so like peer-reviewed scientific literature, but even that, please question everything. And I think that's like the main point of this. Please look up every single thing I just said and double check it. Please, and then if you find something I said wrong, tell me. Because this is all a community and we should fact check each other, and it's all a collective growth. But it boils down to money, it's cheaper, so.
Yeah, I just want to say thank you very much for everything you said, and you hit a lot of really high points. The notion of this mycelium versus fruiting body, all this stuff is kind of moot to me in a lot of ways. We also, there's a lot of equating, when you talk about oxygen, fungi don't breathe, they don't have lungs. Plants don't breathe, they don't have lungs. We don't have gills. These are different organisms, entirely different, older. We are more like them than they are like us. And around that as well, it's if I want to get a lot of tannin in my body, I'm going to eat the roots of an apple tree. But if I want to enjoy my time, I'm going to eat that fruit. And that's what it comes down to, ultimately. And it's, what you put in a product is what you get out of it often. And that being said, wild harvest tastes a lot better, but you need to do that responsibly, and you need to be aware of where you're getting that from and how you're getting that. There's a lot of issues with chaga harvesting, and that interaction of the organism, the fungal organism with that plant as a parasite, and this immunoresponse, that dynamic chemical action is what's creating these compounds. That's great. Everything occurs in different ways, but you need to do it responsibly. Also, mushrooms are phenomenal bioaccumulators. You get a lot of heavy metals from them on top of the nutrients, so you got to know where they're sourced from and how it's sourced. I don't consume a lot of functional mushroom products or otherwise. I'm a mycologist. I don't know, I eat a lot of bacon. But for me, maintaining the organism that maintains the entire ecosystem is much more important. So just keeping that in focus.
That you touched on a ton of points, which I have a longer version of this that I touch on a lot of those things. Chaga sustainability is huge, but also the big industrial cultivation is, can be extremely unsustainable, and we have a long way to go on improving our industrial cultivation techniques to make it more sustainable and better for the planet. We have a long way to go in pretty much every aspect as human beings, even beyond mushrooms. But yeah, thanks for saying those points.
Do these compounds degrade over time? Should I toss my really old bag of
Your new bag of mud water.
Okay. That was two questions.
Yes.
So a year or two, or what's a good time for
I mean, this is beyond mushrooms, but every single product should have a best by date, and so just do that. But I would also use 100% fruiting bodies. Leave it at that.
Is there any or discussion, I should say, in the industry for some sort of certifying body or standards body that regulates it?
For fruiting bodies and mycelium?
Bodies versus, what's actually in the product?
Well, the FDA, I mean, yeah, well, I mean, that's the problem is yeah, I don't know. His question, I don't know why there's not a class action lawsuit, because I think it's 1973 they made that article from the FDA where you have to label it correctly. And so it is, FDA is the agency that is regulating that. And so they are breaking FDA compliance. And so,
Well, for instance, in electronics, there's ULA, which you have to send your electronic products, and then they say, okay, this thing's not going to explode. And then every consumer knows what that little logo looks like. Maybe they're with Novozymes or the enzymatic company that you mentioned, some sort of label or something, some sort of badge that this has actually been looked at by a third party.
yeah, and that was one of the inspirations for having the QR code of like, I want more transparency in the industry, and like, even whatever you label it with words, it's like, I want to see the lab results, which is a whole nother thing of like, there's bad labs out there and things like that. But yeah, I think the more steps that we can take with certifying agencies and transparency with lab testing, the better.
I mean, you just have to look at how the FDA refuses to label GMO. It's because it's money. They want to make the money. They don't want the label correct. And the ones with a lot of money, they don't have to fear anything. Because you can go and put 10,000 lawsuits, they have so much more money for them. And then when you even win, they get a slap on the wrist. What is a $500,000 fine if you make $2 billion? It's in their accounting.
It's all about finding really good mushroom companies like Ben and Amanda with Malama, and there's good mushroom companies that exist out there.
So that you have that one slide that there's differences in your physical response by taking them tincture or tea form.
In unextracted versus tea, yeah.
Right. So
Yeah.
is that, I can infer that it's probably like that for all the different mushrooms have different, and then in capsule form would have a different effect in your body's ability to accept it, or?
Yeah, and then all capsules are different what's inside, some capsules are extracted. It's also personal choice, and I tell this to everybody, like, whatever's easiest to form a habit, great. If you don't like taking capsules, don't take capsules. If you have to like force yourself to swallow it every morning, like, that's not the format for you. If you love gummies, like, go for it. If you love like cacao, like, do it up. Whatever. As long as there are mushrooms in your blood system, like, that's all that matters.
Yeah, just curious around the FDA stuff. Like, do you know how big their panel is, or what they're looking for, like contaminants or presence of specific compounds? You know what that looks like, kind of on just a generic. You get your cordyceps milligrams from X lab, and it goes to the FDA. What are they looking at? Are they looking at like chemical contaminants or just the content, or both?
So you need to prove that it is the species. So you're doing DNA analysis, and sometimes you will have a chemical fingerprint that they are comparing to as well. Other than that, it's pretty basic in terms of heavy metals and microbial contaminants, but not in terms of mycelium and fruiting bodies. That's only a labeling concern. Yeah.
Kind of building off that, are you aware of use of pesticides or any problematic chemicals in these really large-scale farming operations you visited? Or do you foresee that being a problem in the future as this grows?
Not really. I'm sure it exists, and this is, when it comes into certifications of, it's not perfect, but having USDA certified organic certification will help prevent that. It's important to know that once you have that certification, it also means non-GMO, but having the non-GMO doesn't mean it's organic and pesticide and chemical free. And some people think they're the same thing, and they're not. Like at that butterfly, non-GMO project, a lot of people, they're like, oh, that doesn't mean organic as well. And it's like, no, that just means not genetically modified. So, yeah, I don't know if that answered your question.
I guess as you're talking about this industry growing, maybe beyond small companies, you see the videos, the pictures you're showing in China, they look like industrialized agriculture. In Hawaii, we have the issue, scientists are doing a lot of experiments with pesticide-resistant products. I just wonder if this is something that, as the industry grows, might contribute to harm.
Potentially, I haven't heard anything of. Yeah. It's really. No, I haven't heard it. But yeah. Yeah.
I was wondering, is your mycology tutorial available online or in any way?
Tutorial?
Well, you talked about doing this from kindergarten to
Oh no, that was a university in China. I wish I was a part of that. No. But there's a ton of resources online. And Fungi Foundation is also working on teaching kids in classrooms. I have some YouTube videos. There's tons of YouTube videos out there. Tons of books as well. So, yeah.
Someone here last night who asked about Parkinson's at the end of the philosophy talk. I didn't have anything, but you mentioned the rugidacine.
Yeah.
Do you have a paper you can send me about that?
It's a hypothesis. So this is like what's it called? It's like wood lovers paralysis or something like that. In some like in wood growing psilocybe species, some people they have this phenomena where they like can't move their body, like sleep paralysis. And it kind of freaks some people out, but I was talking with some researcher and he was like, "Well, this could be awesome for people with Parkinson's and they have these tremors of like if that could just stop, they could be like, "Oh, I can't move my body. Like this is great," you know? So that was his hypothesis. It needs more research. But yeah.
Alex, forgive me, you might have already said this, but is your book available to buy now?
Pre-order. To buy on May 2nd.
Okay.
Yeah.
How do we do that?
Mushroomrevival.com or pretty much anywhere. Barnes & Noble, Amazon, all the stuff.
So you said they found some 69 species under the seafloor, and they brought it back in that they're ancient life forms. Did they find what did they find exactly?
The spores. So the ones that I showed on the slides were spores of a schizophilum that they germinated on a petri plate.
Underwater spores.
But that's a good question. I have no idea.
Sea mud and examine the sea mud, maybe? Well, I mean, that's
Do it up. Yeah, make it your focus, man. You'll be come back next year. You'll be the marine fungi expert. Let's go.
You're also talking about spores. Schizophilum is one of the most common fungi on the planet that we know of. And it's kind of like saying, well, you found these ancient ocean seeds. How'd they get there? Like, they traveled. That's what they do. That's what they were evolved to do. So it's like they probably came from something terrestrial. They were on land and they were subsumed by the ocean bed. Like, you know, who knows how long they've been there.
There's this concept in Chinese medicine called Du Yao. It's like a pairing of two.
Uh-huh.
Like, Dong Fai Dan Chen kind of thing. In all your exposure to cannabis production and TCM, was there a pairing of cordyceps with any other mushroom?
I also studied TCM for a few years, and really any practitioner that I've ever talked to, every piece of literature that I've read, pretty much all functional mushrooms can be clumped together, and they pair really well together because they share a lot of the same compounds, like 1,3,1,6 beta glucans. They're all pretty much adaptogenic. They all kind of share similar things, but if you want to go more specific, I haven't really heard much literature on like two specific pairs being really good. But like I personally really like lion's mane and cordyceps together, or lion's mane and reishi together before I go to sleep. One really interesting pair that I've had a couple friends do is cordyceps and psilocybe cubensis, which in my experience and all my friends' experience, it increases the effects of the psilocybe. And I think there's some beta carbolines in Cordyceps militaris that's increasing the effect. There's the same beta carbolines in Ophiocordyceps senescens as in Psilocybe cubensis, cordycynine C and D, that are MAOI compounds that increase the effects of psilocybin. So maybe we have some of those compounds in Cordyceps militaris as well. True. Yeah.
Like it is specifically better for ED or, no libido if it's like with other things. Like it always pairs well with others. And then I just have one more short question. Would you say that cordyceps is a stimulant? That's like a debate.
Depends how you define a stimulant. It's very subtle. Yeah.
And would it have any long-term effects, you know, on adrenal function, like being, like when it burn your candle at both ends, everybody?
No, I've read that it helps support yang and jing. And it actually does not deplete jing, it does the opposite.
And adrenals, the adrenal fatigue that's caused by like coffee and other stimulants, like that would be my factor of what makes it a stimulant. Is does it cause adrenal fatigue in long use?
No, it actually does the opposite. Yeah, and actually, a little antidote. I'm not a medical expert, so take what I say with a grain of salt. But from my understanding, the way coffee works is it blocks the adenosine receptors in your brain and kind of suffocates your brain. So you go into a fight or flight response, and your brain basically says like, "I'm suffocating. I need to ramp up," and then that's why you get depleted in the afternoon. Whereas cordyceps actually have cordycepin and adenosine, which goes into the ATP production in the body. So it actually is building on a cellular level instead of just tricking your brain that you're dying and you need to escape. And that's my basic understanding. I could be butchering it a little bit, but yeah.
I just want to throw one thing out there for coffee.
Maybe one more question. Yeah.
If you're experiencing a crash after you drink coffee, it's probably mycotoxin contamination. US has very slim standards on mycotoxin contamination with both cacao and coffee. My question, though, for you is, do you have any experience with the Antrodia genus? It's like
A little bit. Yeah, they're cool.
Yeah, like looking at the functional components, like the electron transport chain.
They're really big in Tokyo, in Japan, in China. They're great. I personally don't have much experience, but they're cool. Thanks, y'all.
Give it up for Alex. Awesome job, man. That was sick. Yeah. Summer was actually a participant of the first annual Hawaii Mushroom Festival, and now is, presenting. So we're honored to have her. Crucial member of the team now. Trying to think what else. Oh, I need to plug the rest of the day. So Summer's going to take us through this first step of cultivation. Then we're going to take a little lunch break. Then Samantha is going to tell us on how she applied this technology, this fungal technology, in a third world country, Uganda, with refugees there, and actually setting up a low-tech mushroom grow operation. Following that, Dave is we're going to get our hands dirty. We're going to start growing mushrooms on banana leaf, some pink oysters. So look out for that. And then we will conclude the day with a metamorpho ceremony utilizing your botanical or mushroom of choice. Leave that for you to decide, but we will have Blue Lotus and Lion's Mane Cocoa available. So that'll be Sari and Natalie, who have that awesome tent over there, herbal smokes, and they have all sorts of cool herbal blends. So, Alex is talking a little bit about psilocybe, which I know some of you may have experimented with. Anybody in here a cop, first of all? Any law enforcement in the house? Okay, we got one. All right. Never mind. No, just, psilocybe or psilocybin specifically has some really profound medicinal uses coming out of universities such as Johns Hopkins, Harvard, helping with post-traumatic stress disorder, end-of-life anxiety, postpartum depression, some of these huge mental health, I don't want to say disorders, but mental health problems that a lot of our world experiences. So psilocybin, which is Schedule 1, unfortunately, it is federally it is the most illegal type of substance out there. Cocaine is even beneath it as a Schedule 2 or 3. They deem it has no medical use despite these studies coming out of renowned universities. But anyway, all to say, it shouldn't be legal, but it still needs to be treated with reverence, right? You don't, especially for anyone who's never experienced it, you don't want to just jump into a full journey without proper preparation and a set and setting. So our final thing today by Sari and Natalie will talk a little bit about creating that safe space for yourself and helping others through it as well if you find yourself in a facilitator or sitting position. So I'm really stoked on that last segment of the day. So just know that that's coming. But first we got to learn how to grow these medicines for ourselves. And just know, some of this technology she's showing you in spawn creation can be applied to any mushroom. Let me just say that again. You can use this technology to grow any type of mushroom, no matter its legal status. But I didn't tell you that, and we don't have cameras here today to incriminate me later. So anyway, without further ado, Summer from Spawn Till Dusk.
Thank you, guys. Hello, Mushroom fam. My name is Summer, and I'm a mycologist over on the island of Maui. Personally, I have a passion for finding and cultivating and propagating any local or endemic culture that I can get my hands on. Professionally, as Ben said, I use that to help out with the Hawai'i Fungi Project. Additionally, I am the co-owner of a organic liquid biological soil amendment company. It's a lot of words. Basically, it's a synthetic pesticide and synthetic fertilizer alternative that we're currently doing pilots with a lot of the resorts and golf courses. So what they were talking about microremediation before, it's proven. Hopefully we're on the edge of a paradigm shift with a lot of these bigger corporations. But yeah, my passion project is Spawn Till Dusk, which is a grain spawn producer and mycology supply. And so today I'm going to teach you about how to make grain spawn. So what is grain spawn? The short answer is it's myceliated grains. But for a lot of people, that doesn't answer enough. Basically, when we say grains, we mean cereal grains, which are oats, rye, berry, wheat, that sort of thing. Oftentimes, animal feed grains. But we'll get into alternatives later. And they are inoculated. Let me get out of the way a little bit. They are inoculated with a mycelium of choice. The reason that we use grain spawn to then inoculate, say, an outdoor bed or totes, is the same reason that nurseries have plant starts. Just like it's difficult to go from a seed to, for instance, the dirt in a random person's front yard, it can be difficult to get it to germinate. Similarly, it can be quite difficult to go from spore to mycelium. So I take the work out of that, and I'm going to show you the process that I go through. So last year, we found this. A couple hundred feet over there, a little bit further, right here on Starseed. This beautiful mushroom is Lepista tarda, we believe. We still want to get it sequenced. But if you were here last night, Chase mentioned he got a spore print. I was itching for it because it's gorgeous. We took it, we germinated it on agar, we got it to liquid culture, then to grain. This, it's a showstopper. And this doesn't even do it justice. If you want to see how deep purple it gets, feel free to follow the Instagram, Spawn Till Dusk on IG. We got it out to bulk. We sent it off to Tropical Fungi Academy, and them and the apprentices, Chase, Dave, and their apprentices, successfully got it going in an outdoor bed, which we then got a spore print from and brought it all the way back home. You can get grain spawn right now online from a lot of different grain spawn producers. And I'm not going to tell you not to do it. I'm a grain spawn supplier. I want you to. But knowing how to capture local cultures and make your own is so important. Through some of these talks, you've heard people mention about how there are some mushrooms that they haven't seen in several years. Lepista tarda isn't here this year. From what we've seen, it grows maybe a month out of the year, and that's it. So if we miss our time due to a droughts which may or may not be getting more prevalent, we can miss out on all the wealth of what this has to offer. Chase cooked it up in a dinner. Apparently it was delicious. We wouldn't have that ability if we aren't capturing that genetic diversity. Additionally, a lot of people have gardens in their home. Wild foraging is great, and there's something to be said for harvesting sustainably, but being able to cultivate is a way to have that medicine or nutrition available at all times, just as you would in a home garden. So, in order to talk about how we do the process, we have to talk about how it happens in the wild. Because what we try to do is mimic that wild process. I got my cute little diagram here. So we start off with a fruiting body, which remember it's the fruit. The organism is the mycelium. This is what comes out, just like an apple on a tree. The fruiting body produces spores, which are like our mushroom seeds. And these spores will land on an ideal substrate, which is different depending on what mushroom you have. Some are saprophytic, like dead, decaying material. Some are parasitic, like the chaga. Some are coprophilic. So it finds its ideal substrate. It germinates into hyphae. The hyphae expand out. They find the relevant gender for them, which is not as binary as it is. It's a whole nother conversation. There can be thousands of genders. For instance, Schizophilum commune. Chaser Dave can shout out how many. 23,000. So they find what's appropriate for them. They form a mycelial mass, which are all of these hyphae interwoven. If you've ever kicked over an old log in the forest and you see that white web, it looks almost like a spider web, that's mycelium. So in the correct conditions, oftentimes it's a high oxygen or high fresh air environment, because remember, mycelium mushrooms need oxygen to breathe. That's how they know to grow out and not in. In the right conditions, they will become more and more dense, and they'll form these hyphal knots, which then become the primordia that he was talking about earlier, which are teeny tiny beginnings of the mushrooms, pins. And then we do the cycle all over again. So this is what it looks like in the wild. This is what it looks like in a lab. Very different. Don't feel any need to write this down. We're going to go over all the steps. And side note, this is courtesy of the Growing Gourmet Medicinal Mushrooms by Paul Stamets, which was my mycology Bible when I was learning, and I highly recommend it to anybody who wants to grow at scale. But I show this because I want to have a contrast to ask the question, why is it so different? And the reason is, when we see the mushroom, all we think about is, oh, there's the spore, it landed, that was the mushroom. And we don't realize that there's a battle, almost, or a race, if you want to view it that way, for ideal substrate. So we see what was the most aggressive, what was the most robust, what was the quickest. But in actuality, there are thousands of spores around us right now. They're microscopic, we can't see them. But as this study points out, for every breath that we breathe, that's between 1 and 10 spores that you're inhaling on average. So the mushroom that you see is the mushroom that won. This is something we want to keep in mind, because that frames the process for how we cultivate at home. There's many different things vying for our ideal substrate, which is what we're putting out. And we want to make sure our culture is the champion. And we'll do that in a lot of ways, most often by just removing the competition altogether. So there's a couple pillars that you need in order to start this cultivation process. The first thing is an environment, a safe environment for you to interact with your chosen culture. In commercial settings, this is a flow hood. concept is simple. You have a blower motor behind a HEPA filter. It's pushing enough pressure that contamination isn't able to get in, and it provides a lot of space for people to work in. I'm not going to spend too much, it's a diagram, laminar flow. I'm not going to spend too much time on this because for most mycologists, this is out of reach. They're oftentimes can be a couple $1,000s before shipping to Maui, which oftentimes doubles it. And that can be a bit disheartening when people say, well, this is how they do it commercially, and this is how it's always been done. But this is the point where I want to bring up my favorite concept in mycology, which is this why you trying so hard concept. So in a lot of other scientific fields, you have a bunch of erudite old men with their funding, and they have their books, and this is how we do it because that's how I was told, and they're asking great questions, they're doing great things. But there's something different about the mycological community in that a lot of awesome, at the very least, technique breakthroughs are happening because of 16-year-olds on online forums who have no money and no space, but have a strong passion and determination to make sure mushrooms grow. So instead of just accepting what they're being told, they ask why. Okay, what we need a clean space. Okay, why this? What if we enclosed an environment, and then after it was enclosed, we were able to, sterilize, disinfect the entire thing? Would that work just as well? It would. Why are you trying so hard? So, still airbox follows this concept. The flow hood, like I said, can run you a couple $1,000s. Still airbox, like $30, and it works just about as well. So the first thing we go over is how to make one, because anyone can make one. It's super easy. The first thing you're going to need is a clear tote, then a hole saw, and PVC coupling, hose clamps, rubber gloves. The tote you want to get, they're offered at a bunch of different big box stores, anywhere you can find totes. The one you want to get often has a blue gasket around the edge, and they're considered watertight. If they're watertight, they're airtight. It'll work. And if you can't get one with a gasket, that's fine. When we're talking about cultivating, we're talking about success rates. And a concept I'm going to drill in on is the fact that nature is dirty. And there is no perfect protocol. Even the best grow organizations out there have rates of contamination. We're just trying to get the highest success rate that is feasible for us. What's most efficient? So you get your clear tote with the clasps on the end and the gasket. You get your hole saw attachment for your drill. It's usually about 4 inches. You're going to cut these holes in the front. A lot of people leave it here, and this is what they use as their still air box. But I believe in everyone here. We don't half-ass stuff. We're gonna go all the way. So the next thing you're going to get is a PVC coupling. I didn't put it on here, but silicone to help silicone in this PVC coupling. Hose clamps and rubber gloves. And it's going to go together like this. You have your clear tote. You have your PVC coupling that's going to stabilize your arms going through, and it's going to be what the rubber gloves cinch onto. Whenever you work, you're going to pull your culture, whatever you're trying to transfer, whatever you're trying to inoculate, into the box first. Close up the box, stick your hands through the rubber gloves, and then you're going to disinfect with your 70% isopropyl alcohol, and you're going to wipe it all down, and now you have an enclosed space that's completely disinfected. I'm going to go to a quick rant about 70% alcohol. A lot of people go for 90% because we're in the iPhone age, and bigger number means better. But it's very it can get a little confusing. Basically, the higher alcohol percentage oftentimes means that it's going to be evaporating faster than it's going to be penetrating cell walls, which is how isopropyl alcohol disinfects. It penetrates cell walls and it basically rips it apart, dehydrates it. It's great. 90%, much better for cleaning technology because it evaporates so fast, but it doesn't do as well as disinfecting. So you got your 70% alcohol, disinfected the inside, you waited a minute for the air to settle, and then you sopped up with your paper towel, and now you can work. I'll address right now a question that I often get is, we see a lot of people online who flame sterilize. Should we put a lighter in here? Don't put a lighter in here. You're gonna have a box of fire, which is cool once. So that's your still air box. Very easy to make, very cheap. This is fundamental to your mycological journey. If you get nothing else, build one of these. So much fun stuff. So the second thing you're gonna need is a safe way to isolate, suspend, and store your chosen culture, which is agar. Some people pronounce it auger or agar. I haven't found the correct way yet. Don't tell me. So, and before I talk about this, I was told in the last talk some people can feel a little bit overwhelmed by all the steps to create all the things. I want to reiterate, you can buy all of this. You can buy pre-poured agar. You call me up, I'll sell it to you. But it is good to know the principles behind how all of this is done. Understanding it makes you a better mycologist. And some people don't have the budget, but they do have the time. And it's fun. So, agar. We got two kinds here. So traditionally, you have petri dishes, which you get from medical supply or scientific supplies. You boil your solution, you pour it in one at a time in your safe box, your still air box, laminar flow hood if you're fancy. And then, once again, this concept of why are you trying so hard? There's somebody on an online forum somewhere went, wait a second, we just need sterile media containers that can hold a small amount of liquid. It's got to be sterile. It's got to be sealable. What if we use ketchup cups? Most things that are made for to come in contact with food are required to be sterile and packaged before they're sent to you to mitigate liability. So this works. I use the ketchup cups just as much as I use the petri dish, and I have about the same rates of contamination, even though the ketchup cups are 1/5 to 1/10 of the cost. Load tech, we love it. With parafilm, side note, with petri dishes, you're gonna wanna buy parafilm because our cultures wanna breathe air. So we wrap these in parafilm because otherwise air can get into our petri dishes. With ketchup cups, we've never used parafilm. The thought process is for short-term storage, there's enough air in these that it supplies the culture for as long as it's needed. I've had these sitting for 6 months at a time and the culture was still fine. So as long as it's not super long-term storage, it works. And I actually have a petri dish and a ketchup cup on the table. Maybe Jared can pass those out so you can see what they look like. And I'm not going to go in detail about the description, but I'm going to leave this up if you want to make your own agar. This is the recipe. Or these are two different recipes. One is made with potatoes, one is made with light malt extract. And there's many other recipes out there. Quick shout out to Mike Tyson, MYC Tyson. He has a dog food agar recipe that he's used with Trametes sanguineus, now Fabosporus sanguineus, the bloody polypore, and he's found that allegedly the color is much more vibrant in the mycelium with that nutrient profile. There's different nutrient profiles. The point of agar is to provide nutrients to sustain your chosen culture. All right, so the third pillar we're going to talk about is how we sterilize anything that your culture is going to come in contact with. So this is the agar, this is the tools that we use, this is the container that our grain spawn will eventually be in. So the most common thing that's utilized is the pressure cooker. There's a lot out there. If you're going low tech, this is probably going to be one of your most expensive purchases at $80. You can pick these up at thrift stores. A lot of people, there are techs, quote unquote broke boy tech, where you steam jars for extended periods of time, and that does work sometimes. But remember, it's not about working or not working, it's about success rates. So if you want the higher success rate, this is an investment that's, I believe, is necessary to make. Another question I get asked a lot is, can I do it in an Instant Pot? Yes, and. So the thing about Instant Pots is they do get up to pressure, but the magic number we're looking for when we're pressure cooking our tools, and we might go into this later on, is 15 psi. Now, Instant Pots can get to 15 psi, but what they don't tell you is Instant Pot's pressure fluctuates. So is it completely sterile? People argue. Some say it's close enough for government work. The Instant Pot? So the pressure cooker? What you're autoclaving? As big as you need to put whatever you want to put in there. You can have small pressure cookers, and if you're pressure cooking grain spawn, if you're pressure cooking agar, it can be one of the smaller sizes. But they make pressure cookers that are huge. Yes?
Can you use the Instant Pot for food as well, and then don't worry about
Yeah. Yes. So that's the yeah. So the reason I can go on a quick sort of rant about sterilization versus pasteurization. As I said, it's all about success rates. Whenever you're using a pressure cooker, the purpose is to kill everything from, bacteria and viruses and mold, anything that could exist. It's the pressure and the heat in combination that just destroys everything. When you have just heat or you don't have a long enough time, you're risking having more contamination in your area. But yes, considering it kills everything, you can put whatever you want afterward.
A note on pressure cookers.
Yes.
From a young age, when I was poor, single parent too, in
Yeah.
college, I love pressure cookers, and I found that I could find them at secondhand stores.
Yeah.
Often it's the seal that's broken.
Mhm.
That's the reason people got rid of them. But you can buy that seal. Look up the model and go to the manufacturer for like $2-3.
Yeah, so in case you didn't hear that, he was saying a lot of times you can get pressure cookers for next to nothing because the seal is bad and you can't use it without the seal, but you can buy the seal very cheaply. And that's fantastic. Also, big side note, very important. If you use a pressure cooker, please look up the manual. Please. A lot of people go out and they're into mycology and they didn't have, grandma show them how to use it and they accidentally make a bomb. When you have tremendous amounts of pressure in a potentially enclosed environment, very dangerous. Look up your manual online. Some pressure cookers have a safety valve that needs to pop up before you start pressure cooking. Some want you to wax the gasket every once in a while. It doesn't take you 2 minutes to look up the manual. Make sure you're doing it correctly so that you're not creating a danger in your household. So now we have the pillars. Let's talk about the doing. So you go outside and you find a mushroom you want, and you want to collect the genetic material. The quickest, easiest way is make a spore print. If you do nothing else in this presentation, make a spore print. If you don't know what to do with that spore print afterward, mail it to me and Chase. We'll take care of it. Spore prints are very simple. You take the cap, or the pileus, of the mushroom, you twist that off or pull off the stipe, you put it on a piece of paper, you put a bowl over it. That does two things. One, allegedly, the humidity helps trick it into sporulating. Secondly, and more importantly, remember those spores are microscopic. So if you're seeing them, there's thousands of them there. There's more, and they can blow away on a gust of wind. That's what they're built to do. So by putting the bowl over the cap, you're making sure that no wind is accidentally blowing your spores away. A quick pro tip on the spore print that we found. Remember I said earlier, nature is dirty, which is great, but we want our champion to win. We found that suspending the cap just slightly often decreases the chance of contamination when we move spores to agar. So not a great amount. If you do too high, some mushrooms sort of make their own wind when they sporulate, and your spores are going to be all over the place and you can't see them. But even using toothpicks or straw, something to that degree, is good enough that it keeps the cap, which may have bacterium or what have you, thank you, Keisha, on it from getting on the spores itself, so whenever you do the transfer, you're not grabbing that contam as well.
You mean the cap of this lifted slightly?
Sorry?
What cap are you using?
The cap of the mushroom. Yep. So, see the mushroom cap? Put it right there, right on top. Yep. No, I believe that is the stipe. Mm-hmm. Usually when we suspend it, we have like 2 toothpicks that are flat on the paper, and it just barely suspends it at the cap. Or 2 straws or 2 tiny pencils or crayons or whatever works. Exactly. That's the other half of Spawn Till Dust, by the way. It's my partner, Jared. Hi, Jared. So, next way we're going to save our genetic material is cloning. So spores, he was talking before about spores and like how can they last under the ocean. Spores can last in space. I read at some point spores in a sterile solution are viable for a couple years, and then I used spores that were in a sterile solution like 6 to 8 years past they were originally made. And there's, as I said before, millions of years. Like you can still, they're one of the most, robust things that exist. That's why they believe in the whole panspermia theory, is that they can last in space, they can do whatever. So spores are great for long-term storage of genetic material where you don't have to do much. Cloning is where you get a little bit more fancy. This is where you're going to use your agar. So you made your agar, which was the recipe that we had before. You mixed it up on a stove. You used a media container of choice. Some people use alcohol bottles stuffed with polyfill. You put your solution in that. You autoclave it or pressure cook it for around 30 minutes at 15 psi. And then you take it out in your sterile area, and when it is cool enough to the touch, you pour it into your agar dishes, and you wait for it to cool. It'll form a hard, almost jelly-like substance. So the reason that we use agar is not because it's the most nutritious thing in the world, but because it provides enough nutrition for things to grow slow enough that you can isolate different cultures. So with cloning, now that we have our agar dishes or ketchup cups, we go out into the wild, or we bring our mushroom home, is the most common way. You put it in your still air box. You make sure that you disinfected everything with isopropyl alcohol, up to and including the outside of the mushroom. Because what we're going to be doing is taking that mushroom and ripping it open. Now, nature is dirty. However, the inside of that mushroom hasn't been necessarily in contact with all the things that were crawling all over the outside, so much less chance for contamination. Still high chance for contamination, but we're talking success rates. So you've ripped your mushroom open, you've used your scalpel to cut a little piece of mushroom out, you've placed it on your agar, You've sealed up your agar again with your parafilm, and then you wait a couple weeks. And next thing you'll know, you'll see mycelium, hyphae, growing radially from that specimen. Additionally, most often you will see some form of contamination. You'll see a mold, a bacterium, yeast look really slimy. The most common mold is trichoderma, it's green. The mycelium is always going to be white and usually filamentous. So it's long strings radiating out from the middle. If you see your contamination, what you're going to do is you're going to cut out the mycelium, that filamentous white stuff growing from the middle, and you're going to transfer it to another dish of agar that is clean. And then when the mycelium grows further, you have a clean culture with no mold or bacteria. Sure.
I'm not seeing this. So you build your medium agar,
Yeah. You have your petri dish or your ketchup cup.
and you sterilize that. You pressure cook those, right? Or those come sterilized. You don't have to worry about them.
If you buy pre-poured petri dishes, they're going to come sterile. And the outside is dirty, but that's fine because when you put it in your still air box, you're sterilizing everything that's on the outside of that with alcohol, but inside the box. And then you open it up, and then you just take a piece of that mushroom. And there are arguments. Some people say you get the most aggressive mycelial growth from the, where the gills are, right above the gills. Some people say that you get the most aggressive growth from the butt of the mushroom, down at the bottom of the stipe. I've pulled biopsies from all different parts of the mushroom, and anecdotally, I haven't seen an extreme difference from what grows more aggressively. So.
What about the maturity of the specimen? Does that seem to affect your vigor?
Yes. So I tend to find that some of the younger specimens tend to have more vigor, but that doesn't mean you should only rely on younger specimens. I've had really old specimens. I've had specimens that I thought were done, that looked dried, and then.
Rehydrated anything that's like a dehydrated mushroom for cloning?
Yeah, and find Chase, because he's got a paper that I'm super into about long-term storage of dehydrated, sterile. Yeah, harass him after the class. Yeah, rehydrating mushrooms and then using them. You can have them at any stage. I probably wouldn't go that young, but it doesn't really matter, all stages, yeah?
How would you pick the specimen, the fruiting body that you want to clone versus another one?
I hate to say it, oftentimes when we go out and pick, there's not too much options. I'll just grab what I can. But oftentimes when I'm choosing for spores, for spore prints, I tend to go ones that are a little bit younger. The ones that look older, when you see the edges of a cap and gill mushroom, when the edges start curling upward, it's usually it's sporulated a lot. They sporulate over time, but there's like a normal distribution curve. It's like a little bit, and then there's more. So I lean towards the younger ones. And then the ones that I get spore prints from are oftentimes the ones that I end up biopsying as well. So now we're getting similar genetic material. Yes. Well, I'm going to answer some questions. Give me a very short period of time, so we might have to get some at the end. Also, terrified of public speaking, but thanks. But I also never shut up about mushrooms. All my friends will tell you, I'll just sit there and follow people around. So after this class, find me at any point, and I would be more than happy to help and answer all the questions. Yes?
So I found my specimen. It's super cool. I've got a really sweet scalpel in my clean kit.
Yes.
And why aren't I just using that to cut it open and then just scooping something off the top of the cap or otherwise?
So the reason I don't use the scalpel to cut open the mushroom is because oftentimes if there is some sort of bacteria, when you spray the outside of the mushroom, it is sort of disinfecting it. But if there's a layer of stuff, it's disinfecting the outside of that layer. If you then cut through it, you're grabbing some of that contam and then putting it down into the mushroom that you're then cutting from, if that makes sense.
So you're cutting it first.
Tearing it first. tearing it first and then cutting from inside the mushroom.
You tear it, sterilize it, put it in there, cut it.
Sterilize the agar, disinfect the mushroom, tear, cut. Yes, yes.
I really want a scalpel and a tweezer makes a baby.
Yeah.
Have you ever seen that? I want a pair of tweezers that has tips that are as sharp as a scalpel.
So there's something what's hilarious is I did not think I would ever need this prop, but there's a set of scissors here. There's a set of scissors here, and that we use oftentimes when we're using scalpels. I'm prone to cutting myself when I'm messing with them. So Jared gave me these to save my fingers, and they're yeah, right. And they're tweezers at the end, and then they have little cutters back here. Yes. So that's as close as I can.
Can I check those out later?
Yeah, hey. They're not sharp on the end. Thought I was gonna murder my boyfriend on stage. All right, so we've now cloned our culture. We've got it grown out. I'm gonna very quickly gloss over this. A lot of people want to know how to make liquid culture. It's advanced. It's easy to do, but that's the problem. If you do one thing wrong, the difference between liquid culture and agar is liquid culture is that 2D space. You can see the different contaminations. You can cut your mycelium and transfer it away. Side note, never cut out the contamination. A lot of people want to do that, but you don't know what has sporulated in there and what may continue growing. Always transfer clean mycelium away. But yeah, that's the 2D space you can isolate. This 3D space juice, you don't know what's growing in there unless you have a microscope to test it with. But I've seen horror stories where commercial growers make a ton of liquid culture that looks fine, and then they inject 100 bags, and there was a little bit of contamination. That little bit of contamination got into all those bags. It's all done. So a little bit more advanced. However, I do mention liquid culture because this is how you're going to get most of your mycelium cultures. Anybody who goes online and searches for pink oyster culture, blue oyster culture, lion's mane reishi, etc., you're going to get it in a little syringe. Your little inside is sterile media container, outside is dirty, that you're going to use in your still air box. And that's going to be liquid culture. It's just a nutrient broth to make sure that mycelium keeps growing. And these commercial farms that produce these liquid culture syringes are usually extremely good on their sterile procedure because they get yelled at if you buy it and there's contam. So and it's also really easy to transport. So liquid culture is how you're going to get your syringe. That's usually how you're going to inoculate whatever your substrate is. All right, so now we're on to the main bit, making the grain spawn. So last time I was told I was a little clinical. So this time I'm not gonna scare people. I'm gonna tell you the easiest way that you can create grain spawn at your house with no special tools. Nothing. You need 4 things, 3-ish things. An Uncle Ben ready brown rice, medical micropore paper tape, and then alcohol and a paper towel. Now, some people, I would prefer you did this in a still air box, but I've seen this done in open air. As long as you don't have a fan above you, still enough air, and people get away with it. So this is as simple as you're gonna get. You can go home tonight, order liquid culture off of the internet, and do this. It's fantastic. So I brought a little, can I walk over there without the amp yelling at me? Thank you. So Uncle Ben's 5-minute ready rice, it comes in this package. It's not the box, it's the package. And you wanna make sure you're getting whole grain brown rice. The white rice does not have enough nutrition. The medleys, the cheese and herbs, it all kind of messes it up. You wanna make sure you get whole grain brown rice, 90 second mycology. on the way here, we couldn't stop at Walmart because Walmart is evil. We stopped at a Whole Foods store. They have an alternative. By the way, that Uncle Ben bag that's fruiting, that's not mine. That's Samantha's, who's speaking. Yes, incredible. So that amazing treasure comes from that treasure. Just making sure everybody knows. So yeah, you're going to take your Uncle Ben's rice bag. You're going to cut. Hey, do you want to demonstrate, Jared? You want to try?
I do it a little differently. I don't even cut. I just take the syringe and you take your bag. So you got your liquid culture in here, and then sterilize.
Do you want to?
Yeah, you want to mic me? So you would sterilize the outside of the bag, or at least disinfect it with some alcohol, like an alcohol wipe or paper towel or whatever, and then you can just inject straight into the bag, usually about like the upper third, usually like a cc, 2 cc's per.
You want the upper third because if you get like into the rice, sometimes you can have difficulty.
Yeah, and then oftentimes I won't even use a separate cutter. I'll just take the same syringe and just make a couple of extra air holes. And you just want to make sure that any holes that you make into the bag, you cover up with the micropore tape. And that way, because as it's colonizing, there's going to need to be an air exchange. And so you want any air that enters the bag to be filtered so you don't get spores and contaminants.
Thank you. So yeah, as he mentioned, and some people do it differently, but the basic process is you want to cut a hole for air. He uses the syringe itself to cut holes. There's back and forth about how much air exchange is needed. I would recommend, if you're starting off to be safe, cut a little hole. You can oftentimes use a little hole punch, pull the front out, hole punch it, and then you use that micropore tape, slap over the holes. Now, the inside is still sterile, but air can go in and out through that micropore tape. The micropore stops the contaminants from getting through, but the air exchange still can. Sure. Yes, medical paper tape, not the fabric kind, not the plastic kind. Micropore medical paper tape.
Micropore a special kind of medical tape or any kind of medical tape?
So medical tape often is micropore. So it's called micropore because it has tiny pores that don't allow contamination to get in. So this is the kind you want. And then micropore tape. That's gonna, whenever you cut your holes that allow for the gas exchange, put that over. And then as he mentioned before, you can take your syringe, you inject it in. As soon as you pull that syringe out, slap some micropore tape over the hole where you injected. And then, yeah, just like you see here, once you feel it fully colonize, it'll get hard once the mycelium, or harder once the mycelium starts taking over. You can take that grain and use it like any other grain spawn. You can mix it into a tote to bulk it out and grow more. You can, throw it in your garden bed. Some people just cut off the top and fruit straight out of the bag. It's so easy. You don't need a pressure cooker. You barely need anything. Yes.
It's clear on the bottom of the bag, so you're supposed to use that. But it still looks like rice, but the top is hard.
So that's the thing. If you inject the bag, he's saying if you inject it and there is a clear port on the bottom. I'm going to show, but I don't want to dump that out. There's a clear port on the bottom. You can see the brown rice through, so you can see if it's fully colonized. But he's saying, what if it's hard on the top and you don't see anything on the bottom? Time. So one of the things that happens in the wild, right? You have these different phases of time. You have when something originally germinates, and then when that mycelium is growing, we call that the spawn run, and it travels throughout its substrate. Usually, the conditions for it fruiting are oftentimes, has it fully colonized the thing that it's in? We don't know every single factor of what causes things to fruit. Morels, people grow morel mycelium all day long. How do you get them to fruit? So same thing. If the with oyster mushrooms, which is what is most common, that's our strongest champion. That's the quickest, most aggressive thing we can do. That's often why it's the primary thing we work with. If it's grown and you don't see it on the bottom, but it's been like 2 weeks, you can probably open it up. It might be that there's too much water sort of on the bottom.
Can you do a break and shake with Uncle Ben's?
So Jared's talking about a break and shake, which is sometimes when you if it's sealed on the inside and it's sterile on the inside, I mean, it's not too much more that you can do. You don't open it up. What a break and shake is you inoculate something and it's partially myceliated, and then you break that up because the mycelium, every piece of it is like part of the whole. So you break it up and then you mix it with all those unmyceliated grains, and it grows a lot quicker because it has more sort of surface area with the unmyceliated stuff. You can do that with Uncle Ben's, and that sometimes works. So something else, if you're doing a hole punch or if you're cutting, don't cut straight. I say don't cut straight through the middle. Don't hole punch straight through the middle, because oftentimes what people will do is they'll put their micropore tape on either side and then it sticks together and then there's no air exchange. Yeah, so little quick pro tips.
So the Uncle Ben's is dry, isn't it?
It's not. So remember, the same concept. Why are we trying so hard? Some kid somewhere was like, hey, you know what? We need a sterile space. We need a grain that has the right nutrition for our mycelium, and we need enough water to hydrate that grain, because the mycelium needs water as well, just like we do. And they looked at the Uncle Ben bag and they said, wait a second, it's made for food, for consumption. It has to be sterile in there, because otherwise lawsuits. And it has the grain that's sterilized, and there's enough water in there, because the point of Uncle Ben's is you're supposed to microwave it, and then the water is forced into the grain. So that's where the water comes from. And to your question, the only time I've really seen it not colonize the bottom is in really, really old bags where water is sitting in the bottom, and then the mycelium doesn't want to drown and doesn't take over that. But try to use fresh Uncle Ben bags rather than old ones. So that's the easiest thing. And then the most common way that, am I good on time? Okay. The most common way that people create grain spawn is jar tech with mason jars. And the concept is you have a mason jar, you have your cereal grain, which we will sterilize in ways that we'll get to in a second. And then you have a modified lid, because we need a way that it has to remain sterile, but we need a way to introduce mycelial culture, and we need a way for that mycelium to be able to breathe. So this is how you make a modded lid. The most common ways these modified lids are made is people will oftentimes use like a drill, and they'll drill 2 holes. In one hole, you can buy injection ports at medical supplies or online, and same thing with gas exchange ports. It's the fancy way to do it. You drill the correctly sized holes, and you can push those in. Bam, you have everything you need. You sterilize your substrate, put that on, done. Low tech, because we love our low tech. The reason for the injection port, it's called a self-healing injection port. It's whenever you stick your syringe in, you're creating that hole. That's a vector for contamination. But once you pull the syringe out, if it heals itself, it isn't a vector for contamination, because nothing is getting in and out. And a self-healing injection port is so dense that when you force it apart with that syringe and you pull the syringe out, it goes back into place. Same thing, why are you trying so hard? We got high temp silicone, does the same thing. Make sure it's high temp, because otherwise whenever you put it in the pressure cooker, it's just gonna melt. It's not gonna be good. But you get high temp silicone, you drill or hole punch a hole in your mason jar lid, covered in high temp silicone, wait for it to set. In your other hole, you're going to take polyfill, and that is pillow stuffing. And it's so dense that, similar to the other micropore stuff, you're not letting contamination in. It's much cheaper, arguably. So that's how you make these modified lids. So the marble and the stir bar is actually, these are the ingredients actually for when you're doing, you don't need these unless you're doing liquid culture. So when you're doing liquid culture, you put a stir bar in the bottom, so when you sterilize it, it can stir the culture, and the marble takes that place. But if you're just doing grain spawn, you don't need the stir bar and the marble.
Are you poking the polyfill into the hole with like a needle or something?
You don't necessarily need to poke a hole. Like I said, you make the hole that the polyfill goes in with a hole punch or a drill, and then you can stuff it in with your fingers because all this stuff is going to be sterilized.
So the hole is bigger than a pin. It's more like that size.
You can make it a good size hole, yeah. I've seen people use polyfill in big shotgun totes, and there's like giant holes that they stuff in, and it works. So, yeah. So yes, polyfill or self-healing injection ports and gas exchange ports. Those are your modified lids. And then you have your mason jars. And now you have to pick your grain. So there's a couple different reasons people may choose different types of grain. In the commercial space, we often use wheat, rye, oats, barley. I personally use oats in a lot of my sterilized grain. If you're buying from me, you're mostly going to get oats. So when I say these grains, this is not something you're going to get at a grocery store. You're only going to get it from an animal feed store. And the reason why is the stuff that's for animal feed has hulls on the outside, and that helps protect the inside from contamination. But the bacterium loves that super nutritious, wet stuff, and the holes on the outside sort of protect that. When you sterilize it, it's still technically kind of hard on the outside because of those holes. So animal feed stores is where you're going to get this stuff, not grocery stores. One of the other major factors people think about when they're choosing what grains they want to get is inoculation points. Things like millet are very small little balls. They have a lot more surface area. Thank you. They have a lot more surface area whenever you break it up and you want to inoculate something else. If you have less things that are bigger, the myceliation is going to happen in those places. For instance, if you had this would never happen, but if you had two oats and you threw them into a bag, they're going from those two places. And if you have 5 pieces of millet, which is occupying the same amount of space, now there's 5 different places that are going to be myceliated from. So smaller, more surface area. Some people combine the two. That's something we're moving into, is combining oats and millet to have that combination of oats are hardier, they're more forgiving. In a pressure cooker, they don't burst. If you have a bunch of burst cereal grains, vectors for contamination. And then cost is another huge factor in why people choose the grains. A lot of people successfully use wild birdseed from the hardware store, and it works. Colonization times may not be as quick, but it works.
But you have to take something out like
Sorry?
sunflowers and use them. You have to take those out, I guess.
I haven't seen people do that. I've seen them just use the whole thing. And that's a fun thing about mycelium. They've done texts before that have they grow on cardboard, and people ask about the staples or the tape. And the mushrooms don't care. If they won't eat it, they won't eat it. They'll just grow over it. So I'm assuming the same happens with, our birdseed. So now that we picked our grain, we have to figure out how to sterilize it. And there's 2 main schools of thought, which are in big contention. It's like the Apple versus Windows debate. It gets very heated on these online forums. And it's to soak or not to soak. And The reason why people say to soak your grains, A, is because it sort of starts pre-hydrating your grains. And also there's this thought of endospores. So some fungi, a lot of bacterium, if they're not in the correct environment, they will go into this dormant state where they're extremely hardy in extreme temperatures. The thought process is if you soak your grains, you are germinating those endospores. So whenever you sterilize them, you're killing them. But there's an argument that you may or may not be killing them anyways in the sterile cooker. The big thing that the big reason that I've seen for doing one or the other is time and aesthetics. So with the soak method, you're going to take your grain. Oftentimes, you're going to put it in a tote. You're going to soak it overnight. Once it's done soaking, you pour off the excess water. You take your grains, you simmer them for like 30 minutes. And then you fill your jar. You drain them, and you want to make sure they're dry enough on the outside. There's this thought of field capacity. You don't want to overhydrate your grains, because remember, if it's too wet, the mycelium won't want to grow there. But you don't want them to be too dry, because the mycelium need a certain amount of water. So you want to follow the rules on how long. So soaking overnight, boiling for 30 minutes, that's going to put enough water in it. And then you're going to fill your jars. Put your
You skipped the drying the grain out part. Like if you have your fully soaked wet grain.
Yes.
How do you dry your grain out?
So it depends on how much grain you're doing. If you're doing a ton of grain, some people make giant sieves out of, they create a wooden frame with pieces of wood nailed together, and then they put window screen or insect screen on the back. You can dump your grain on that, leave it outside on some cinder blocks for a little while. It's dirty, but we're going to sterilize it anyway. Small batch, small batch. A lot of people just lay it out on a baking sheet and just wait. Put paper towel under and over it, and then once the paper towel has just a few drops of wetness, it's not soaking, they say, okay, it's good enough. They fill their jars. This is once again a good time to reiterate: oats are very forgiving if you under or over soak slightly. And then the other big process is no soak, no simmer. So thought process behind that is instead of soaking and simmering, we're going to put in the amount of grain, about half the amount, because oftentimes it'll double in size. And then we're going to put in the amount of water exactly that will hydrate that amount of grain. There's a lot of different recipes online for what that water to grain ratio is. You want to make sure you get that ratio right. I have one here for millet. Feel free to write down. If there's another grain of choice that you like that you want to do no soak, no simmer, look it up. There's tons of different recipes.
What's your practice? Do you soak or not?
I hate to say this, but soak, I kind of have to do the soak and simmer because, as I mentioned before, aesthetics. In the process of soaking and then dumping that water off and then boiling and then draining it, any weird seeds or rocks or dirt that oftentimes comes in animal feed bags gets sort of washed away in that process. With the no soak, no simmer, even though it works just as well, if I hand my customers a bag and it looks kind of dirty, I'm like, I don't trust you, even though it grows just fine. So aesthetics is one of the reasons that I don't do no soak, no simmer that often. However, I have done it, and once again, anecdotally, I've had just about the same amount of contam rates between no soak, no simmer and soak and simmer. And no soak, no simmer takes a fraction of the time. So you just pour in exactly what you need, sterilize. The sterilization process, that heat and pressure, forces that water into the grain. So
do you not use gypsum for the soak method then?
I do not, no. Some people do. So there's a lot of different supplements and amendments people talk about utilizing when they're making their grain spawn, and they all have their advances and drawbacks. Once again, anecdotally, it's different for every person. I've noticed that the amount that I've used, I haven't seen a drastic difference in germination rates and myceliation rates between one or the other. Same for no soak, no simmer. And when you're talking about And it also depends. So a lot of commercial growers will use what's called master's mix, which was pioneered by, I believe, Earth Angel mushrooms, which is it's a bulk substrate that's half oat pellets and half soy hulls. Tremendous growth, different. Other people say use coffee grounds in your bulk substrate. Use coffee grounds in your grain, higher nitrogen count. And then I don't see the difference. So do your own studies, like they were saying. If a 16-year-old on the internet can figure out how to do ketchup cups, you guys can go, especially when all these resources are fairly cheap, try it out and then call me. Send me an email. Let's figure this out together. People like us are making breakthroughs, not just in general mycology, but in these techniques every day. And then, once you have your grain, you filled it with your cereal grain, you pop your modded lid on, it's hydrated. You sterilize it for about an hour and a half at 15 psi. Now it's sterile. It's sterile inside the jar. You take it out of the pressure cooker in the dirty air, but it doesn't matter because inside the jar is sterile. That environment is perfect. You bring that in your clean environment, in your still air box, with your liquid culture and everything else. Then you disinfect the outside of everything. Now you can take your liquid culture syringe, inoculate that grain jar, wait a couple of weeks. That thing will fully myceliate. You'll see it's going to be pretty and white. And you can use that to inoculate your garden bed or your totes or whatever else you want to grow with any mushroom culture you want. I'm not telling you what to grow. Any mushroom culture you want. And then really quickly, and then I'm going to finish all the questions. Some people will inoculate, they'll make a grain spawn jar, and they'll say, okay, this is it. A lot of commercial growers, and Stamets himself in his book, says the ideal way to be most efficient financially, you can take that grain spawn jar and inoculate more sterile jars with it. So instead of using a liquid culture syringe, oftentimes those can run you, $10, $15, whatever. You can take there you go. He's got one. You take your grain spawn jar, you sterilize a ton of other grain spawn jars. They're all sterile grain. You pull it into your still air box, and you use that myceliated jar, break it up, put a little bit of that mycelium in each of those jars. And they call that generation 1 and then generation 2. And you can sort of keep expanding that out. The only thing that stops you is this concept of senescence, which is super fun. It's growing old in all things. It's the concept that every cell division we get past, from the haploid to the diploid, every cell division, you lose a slight amount of vigor but you become more prone to contamination. So if you expand out too far, if you were to take one grain spawn jar and then fruit it, and then break that grain spawn into another grain spawn jar, and then fruit it, and then break that into another grain spawn jar, 10 jars down the line, you might not grow anything, or if you do, it might be mutated. A lot of the mutations people see in these mushrooms happen from these generation 10 gene torturing experiments. So just be careful. But oyster mushrooms, you don't really need to worry about it too much as long as it has enough nutrition where it's going. I've seen oyster mushrooms go for quite a few generations and be perfectly fine. And now I'm done. Questions?
I have a comment. Trad Potter wrote Organic Mushroom Farming. He says that if you change the grain after so many expansions, that it can prevent senescence.
Yeah. So I don't know about senescence necessarily. I would have to look into that. But I know for sure, anecdotally, people have spoken about how going from one substrate to another helps increase vigor at the very least, where it starts losing vigor.
This is true in pretty much every step, whether it's your agro recipe or the grain you're using, so you can kind of play with keeping them stimulated. They honestly get bored. They're like, I'm tired of eating oats, Summer. Give me something else.
Yeah.
So you can just transfer from grain to rye or vice versa.
Yeah, Jimmy. Yes, so that's a very important concept. It's definitely more important if you're using polyfill than if you're using these other gas exchange ports. Polyfill in particular, if you're going to sterilize it in your pressure cooker, put a piece of aluminum foil over that polyfill, because otherwise what happens in that pressure cooker, you might have some condensation that builds up on the lid and then drops into that polyfill, and then vector for contamination. Yes.
You use the small mason jars for cloning. You mentioned petri dishes and the ketchup. But have you used the small mason jars for the cloning too?
I have not, but I've seen it done. There you go. Yeah. Yeah, it's great. Yeah, like why are we trying so hard? Question?
Yeah, for our liquid culture,
Mm.
it's gonna make more syringes. Would we just take what we soaked our grain in and then filter that out and dilute it like 1:4 and then inoculate that with whatever else we have in our syringes and then create more liquid culture to make more syringe?
So usually when you're creating, if you're wanting to create, first of all, if you want to make liquid culture, talk to me after class. That's a whole thing. But we did a whole class on it. She was there. The liquid culture, if you want to make liquid culture, most often you're going to go from a culture in a petri dish. Some people do Andrew Mossy Creek mushrooms does biopsy directly from the mushroom. That's some scary stuff, but usually petri dish culture, you take a tiny little slice of your agar with your mycelium and you drop that into liquid culture, and that's what you use to expand out. Yes.
Okay. Is there any specific environmental conditions or light when you're growing it out in petri dishes?
No. So in the spawn stage, mycelium doesn't care so much about light, and it's less. It needs some air, but it's less strict about how much air it needs as opposed to when you're fruiting. If that answers the question. So yeah, we have all of our cultures stored in the dark at the back of a shelf that we've forgotten about, and they're doing fine. No. If it's light or dark, I haven't seen a huge. Don't put it in sunlight. Sunlight will kill your. Don't put your cultures in sunlight. Unless it's Termites, it's just a beast. But like everything else, don't usually put your cultures in sunlight. And once again, side note for whoever may use this, don't put your. Some people store their cultures in a refrigerator to get it to last longer. Long-term storage should be a very particular. There is a very particular media bottle and how they do it. That's a whole different thing. Some people try to extend the shelf life by putting it in the fridge. The thing I grow the most is Pleurotus jamor, and it's scary to put those hot weather mycelium in the fridge, so that's something to worry about. And patty straw. Chase has given me a bunch of patty straw cultures that I immediately killed by putting in the fridge. Any other questions?
Yeah, I was just curious about if you took a spore print and like saved it in the fridge, what do you think, just kind of ballpark would be still viable to
Spore print? How many million years was that one underwater? So you're good. Yeah, you don't even need to put a spore print in the fridge. I found one under my bed. Like, yeah. Spore prints don't care. Make spore prints. Tyvek suits? Tyvek tops. Like the plastic tops?
like a mason jar ring and just a bit of Tyvek, like your free unrippable postage, and just use that as your
I have not. He said he did. Yeah. Yeah. There you go. Yeah, there you go.
You can even get free tape from the post office too.
There you go. Yeah. I've seen, I won't say what was in it, but I've seen gorilla mycologists that like take liquid cultures and, or liquid culture and fill super soakers and then just go out in the wild and they're like shooting stuff with it. Yeah. Mycologists are awesome. We're way cooler than those other scientists. Yeah. There's, so this process, if you want to see this process, Tropical Fungi Academy is always doing classes, and they're great. They employ me to help teach you how to make spawn. There are further resources online. Like I said, as far as books, Growing Gourmet and Medicinal Cultures by Paul Stamets is fantastic as a book. If you want to see it online, which I do advise, there's tons of YouTube tutorials online. 90 Second Mycology does Uncle Ben Tech. Earth Angel mushrooms, Myers mushrooms, Mossy Creek mushrooms walks you through how to make grain spawn. I'm sure there's a ton more out there. And I'm sure Mushroom Revival has something going on too. Big props to him. Good? All right. Thank you so much. I appreciate it.
Awesome job. One more time for Summer.